All right, good morning, everyone. Good morning. Thank you for being here for Download Day. Thanks. Give yourself an applause. I know a lot of you have traveled. It's hard to make it to Salt Lake City. The number one question I've got asked today is, do we require everyone to climb up the wall? And the answer is yes. So if you haven't seen, that used to be the tallest rock climbing wall in all of the Mountain West. But we are excited to have you here today and welcome you here. It's going to be an exciting day. We have incredible guest speakers coming today. I won't spoil all the surprises, but if you look at your agenda, it's pretty awesome. So we'll leave it at that. And just a couple housekeeping rules before we start the day. Bathrooms behind me to your right, and also upstairs where you were having breakfast. We know that this is a commitment to be here all day. If you do have meetings throughout the day, we do have reserved phone booths upstairs. First come, first serve for that. And if you forget anything that I said, if you find someone with a yellow lanyard on, they will be able to help you. You also have refreshments at the top of the stairs and in the back with the lounge as well. And so without further ado, I'm going to invite our first speaker up, Chris Gibson, co-founder and CEO of Recursion. Chris.
Awesome. Thank you, Ryan. Welcome, everybody. Thank you so much for joining us. I know many of you flew in last night or over the weekend to enjoy the Mountain West, and we're so glad to have you. We've got analysts, investors, even patient advocates who are here for us. And it means so much to have all of you come and join us, our investors as well. So thank you. Thank you for coming out to learn about Recursion. As we dive into the day, I want to just set the stage a little bit for you and share our hopes for what you will take away. And the most important thing is that we want to be able to provide you with updates around all of the key elements of Recursion, our pipeline, our platform, and our partnerships. And we're going to be able to talk today about each of the clinical programs. We have more than 7 readouts coming in the coming 18 months and the months beyond. 5 of those efficacy readouts, 2 of those are going to be phase 1 readouts. And we're just so delighted to be able to start to reap the fruits of all of the work that we've been building on this platform for the past decade. And I think the 7 programs in the next 18 months is just the beginning of what you're going to see in the decade to come. Beyond that, we have incredible partners. We have just incredible partners in Roche Genentech, in Bayer, in Tempus, in Helix, in Enamine, and of course our incredible partners at NVIDIA, who we'll hear from later today. And we want to be able to share some of the updates from those collaborations with you as well. And then finally, our platform. I think unlike many other biotechs, we've been investing in building a platform to help change the pace and scale of drug discovery. That's what we're here to do. And you'll get a chance to see that platform in action today, to hear from the team that's building it, and to understand some of the things that we've built in the last 18 months and some of the things that we expect to build going forward. And I hope we can also help define for you a moment that we believe is happening right now as the world begins to transition from biotech to tech bio. A moment when compute and technology are going to be as core to the biopharma industry as any other tool has been. And we think that moment, which is really years, is happening right now. We're right at the inflection point. We see the C-suites and the boards of large pharma companies starting to take seriously the opportunity that comes with machine learning and AI. And we see companies like Recursion better positioned than almost any other to take advantage of this moment. And we want to convince you today that that is true. And finally, unlike the last Download Day where we had almost entirely Recursion or Recursion-associated folks speaking, this time we wanted to bring in a variety of folks from outside the company who could share with you their perspective, either our collaborators from Genentech or Bayer, KOLs and researchers working on some of the targets that we're advancing, and of course, Ryan, I'll spoil it, Jensen, who's going to be joining from NVIDIA later today. We're so excited to let you hear from them how they see this transition from biotech to tech bio. So I want to set the stage a little bit for the moment that we're in. And in some ways, it's a tale of 2 cities. On the biotech side, we're at the end, I hope, of a 60-year trend in declining efficiency in R&D. This is the number of new medical entities discovered for each $1 billion spent adjusted for inflation. And you can see that it's been going down. And luckily, over the last couple of years, it seems to have stabilized. But it is not stabilized at a good place. The IRR on invested R&D in our industry is not as robust as we would all like it to be. And of course, there are winners in this space. And ultimately, those winners help advance the cause for patients. But I believe with technology, we can do much, much better. And on top of this declining R&D efficiency, we also are facing, going into the third year of really challenging capital markets for biotech. And so there's a lot of headwinds in the biotech world. Contrast that with the technology world, where it seems like every day there's a new headline about a new AI advancement, a new technology advancement. The capital markets are robust behind these companies. There's dramatic increases in compute that are happening. And you really start to see the difference between these two worlds. And I think it's been magnified over the last 2.5 years. But what we see happening right now is this evolution of biotech into tech bio. And we believe, and I think increasingly the world believes, that this transition is not a question. It's not a question of whether it will happen. It's a question of who will drive it, when it will happen, and how it will happen. It is inevitable. And the companies who are going to bring this transition are companies who can work at the interface of tech and bio, companies who are bilingual. And thank you for coming, because you can look around today and you can see what a bilingual team looks like. We have biologists and chemists, some of these folks having spent decades in the biopharma industry, sitting next to machine learning researchers, data scientists, software engineers. This is what the future of biopharma looks like, and you're seeing it today. And companies who invest in generating the data, in creating the automation, and the compute that is necessary to operate at this interface, those are the companies who are going to own this evolution. And we believe that Recursion is better positioned than almost anyone to take advantage of all of these tailwinds. So I want to go back a little bit from this moment to the early days, back to the early 2010s when Recursion was founded. There were about a dozen tech bio companies back then, and each of us started with a point solution. For us, it was this assay called cell painting, popularized by Ann Carpenter at the Broad, and we became the pioneers of industrializing this assay. The entire laboratory that you see to your right is built to generate images of human cells from which we can leverage computer vision and machine learning to extract signatures of what is happening, what the cellular state is. And this new kind of omics is driving a revolution. In fact, today, almost every one of the top 20 biopharma companies is building out these sorts of assays based on cell imaging as a new kind of omics. But we're half a decade ahead. In this laboratory this week, we will do more cell imaging assays than I imagine any of the large biopharma companies will do in the entire year. We have scaled this better than anyone. But at the end of the day, point solutions are not enough. We can discover new targets, we can identify hits, we can even optimize our molecules using these technologies. But over the last 3 or 4 years, we've begun to build the entire stack, the complete vertical. With the same philosophy that we drove to phenomics, we're now deploying across so many different steps in drug discovery and development. And we've earned the resources to deliver this by being pioneers in this space, by being seen as leaders. And today, we're going to be able to tell you about each of these tools, technologies, techniques that we're using, but all of them are based on the same kind of framework. We generate data, we make predictions from that data, and then we test ourselves. And we believe that the companies who can stitch together all of these different point solutions into a workflow are the companies who are going to recognize the compounding efficiencies of technology across discovery, translation, and development. And we believe that Recursion is the company that is furthest ahead in this race. And I hope we can convince you of that today. And we're not just building the technology, we're building ways to interface with that technology. At JP Morgan this year, we talked about LOW, our LLM orchestrated workflow engine, which is allowing a scientist who's not an expert data scientist or a chemist who's not familiar with coding to leverage the entire Recursion operating system to help discover and develop medicines. And as we shared a little bit earlier this morning with our 8K, we just announced that Bayer is going to be the first large pharma company to leverage this tool to help us discover medicines together. That is so exciting. And just to give you a sense of timing, the Valence team showed the early idea for us to us in October of last year. We had a minimum viable product at JP Morgan in January, and now we have one of the largest biopharma companies in the world who's going to be integrating this tool to help discover medicines with us. We think that is what the future looks like. And we've moved beyond just generating data of cells to generating data in animals, to looking at data from humans, and we've begun to start to train models that don't just identify patterns across a given data layer, but increasingly, we are now training foundation models at Recursion that work across multiple data layers. And the reality is no one data set is going to be sufficient to truly decode biology, to decode chemistry, to help us radically improve lives at scale. But by stitching together one of the most compelling sets of data across multiple different levels, phenomics, transcriptomics, proteomics, in vivomics, genomics, patient data, and training causal AI models across all of these different layers, we believe that we will be able to start to extract extraordinary signatures of biology, not just at the gene level or the protein level, but at the organismal level. And if we can do that, we truly believe that there will be a fundamental shift in the pace and the scale of discovery, and it will be companies like us, companies building an operating system, who will underlie that shift. So I want to go back to today and ask the question, where are we in this shift? And I'm going to start by talking about transportation and communication, the horse and the letter. We are in the very early stages of this transformation. Recursion is a little bit like that early car or that early telephone. We are fundamentally changing the way that one discovers and develops medicine, but we have a long ways to go. And in the coming decades, you're going to see this continued transformation, the addition of new modules, the addition of new technologies, new data science techniques that are going to work together in a vertical to help us bring new medicines to patients much more quickly. And over the last decade of building, we've learned a few things. We've learned that fit-for-purpose data is absolutely critical. If you're not building the right data sets in this space, the public data sets that exist today are increasingly being commoditized. People are making the same conclusions off of those data. To stand out, to build a moat, you have to build the data. That is the differentiator. And I don't think anybody else is building it as well as we are. Beyond the data, you have to invest in the compute. We'll talk a little bit later about BioHive 2, our supercomputer, which is just a few miles down the road that we built in partnership with NVIDIA. Recursion, a small public biotech company, owns and operates the fastest supercomputer in the entire biopharma industry. We are making bets where others are scared. This little company is making big bets because we believe that when you bring together data and you bring together compute at scale, you're going to be able to move from very specific solutions to generalizable solutions. Foundation models with emergent features that help us start to understand broadly the way biology and chemistry is interacting. And we're already starting to see those signs in some of the foundation models that we've built today, and it is tremendously exciting for our team. And we're not just building with our own tools, we're even taking tools from the AI world, like LLMs from big tech companies, and we're tuning them to help us do our work more efficiently. If you were to go back to 2022, Recursion had 40 full-time equivalent employees who were working on looking at all of our data and all of the public data to identify arbitrage, places where we had robust signal about some new target or interaction that we thought we could drive to be a first-in-class or a first-in-disease medicine, because that's the kind of place that Recursion wants to operate. And in 2023, we started tuning LLMs to automate this process. And today, in the first half of 2024, with one full-time equivalent, just kind of running the thing and making sure it doesn't break, we have initiated over 201 program hypotheses. We are automating the process of looking at our data, looking at all the public data, and finding places where we have belief that there is something new and novel that we can drive as a potential medicine to patients. And then for very, very small dollars relative to the traditional methods, we can validate those hypotheses, which means we can flood our teams with so many exciting ideas that they can pick the very best ones to drive through translation and into the clinic. And I think you're going to see over the coming half decade the pace and scale of this flywheel as it really starts to turn. And what it also means is that the 39 FTEs that were working on identifying those programs are now working on much harder problems later in the process. So we're becoming more and more efficient. This little team, even though we haven't grown a lot for the last couple of years, is generating more and more value year over year. So I believe we are the leader of the tech bio space. But the question is, how do we stay the leader? How do we lead this revolution of biotech into tech bio? And I believe there's 4 ingredients: people, data, compute, and capital. And I don't believe there's any other team in the world as compelling as the one that we have built here at Recursion. You'll get to meet many of them today. You'll hear from many of them on this stage. But it's not just the software engineers, data scientists, biologists, chemists, people in finance, people in facilities that make this company run. It is our partners. It's our partners at Bayer, our partners at Roche Genentech, who we learn from. It's our partners at Tempus and Helix and Enamine, and of course NVIDIA. And together, this little company with these incredible partners is going to fundamentally change the way medicines are discovered. And the way we're going to do that is through the generation of fit-for-purpose data. You will see today, through this laboratory and laboratories across the street, and our in vivo facility in Southern California, that we are able to generate data at a pace and a scale to iterate on that data in a way that few other companies, even 10 times our size, are able to do. And we've already talked about compute. This little company has the fastest supercomputer in the world. You'll hear today, it's the fastest supercomputer in biopharma in the world. Important point. I saw the lawyers wince just a little there. We've made these investments because we believe the intersection of data and compute is the future of this industry, and we intend to lead it. And this computer has already helped us drive to multi-billion parameter foundation models that we're going to be able to share a little bit more about in a few minutes. Very excited about this. And it's only been up and running for, 6 or 8 weeks or something like that. So really, really exciting times. And of course, capital. We have been lucky to have been able to earn capital from all of the investors who have supported us for the past decade to take this big swing at trying to build something different, to trying to build a different kind of company. And I believe we will continue to be able to earn that capital, but we are on the precipice of moving in the coming years to a new stage for Recursion as we read out clinical assets at relative scale, as we start to sign additional significant partnerships and drive milestones and maybe even royalties from the partnerships that we've signed. I believe Recursion will be entering a more sustainable stage in the coming years where we won't have to rely on the capital markets as much. We'll be freer to build this company at the pace and scale that we believe it deserves. But a huge thanks to all of you who have supported us and continue to support us going forward. So I just want to end with one thing, and that is our purpose. Recursion exists to run an experiment, not the experiments that you see in the laboratories here. It exists to run an experiment to ask whether or not there's a different way to discover and develop medicines. Because despite incredible people across the industry working hard for decades, failing in the clinic 90% of the time is not good enough. Nobody wants that. Not the companies, not the patients. And few people are taking the really big bets to transform the process of discovery, the modalities of discovery. We look back at the Regenerons and the Genentechs and the Alnylams, and we believe there need to be more companies striving to build those kinds of platforms. That's what this industry needs. It doesn't need the 7th or the 8th or the 9th drug targeting mechanism X. And we intend to run that experiment here. And if we are successful, I believe we can have dramatic impact, not only on patients, but on the industry writ large. And that ultimately will be for the benefit of patients as well. So thank you very much. Thank you all for joining us today. We're so excited to have you here. And I believe up next, we have Ryan for a transition.
Transitions are very important, Chris. We'll see you later for a fun Q&A. And now I'd like to introduce Lina Nilsson, SVP of Product, to talk about the Recursion operating system. Lina.
Awesome, thank you. Hello everyone, I'm Lina Nilsson, SVP of Inception Labs actually, one of our research units at Recursion, and I am thrilled to be here today to share amazing work from hundreds of our talented Recursionauts. And specifically, I'm going to give you the inside scoop of the latest and greatest on the Recursion OS. That is the interconnected suite of technical and scientific tools that underpin our unique approach to drug discovery here at Recursion. And after me, you will hear from the awesome Laura Schaevitz, who will show how we have applied the Recursion OS to drive specific drug discovery programs. So building on Chris's introduction, I'm going to show you the how and the why for why the Recursion OS is bilingual. That is science and technology, physical wet lab experiments, and also dry lab in silico modeling. Like much of the biotech industry, we used to do this approaching it like individual point solutions, but we no longer do this at all. We build the wet lab and the dry lab in an interconnected way of virtuous loops. And indeed, over the last year or years, like Chris mentioned, we've really been able to transition this into what I believe is probably the first example of a version of an end-to-end industrialized drug discovery system. And this here is a simplified graphic of what that looks like at Recursion today. Why is this important? One purpose I think is pretty obvious and pretty intuitive. It is if you really want to get better drugs to patients, it's not enough to solve just one piece of the puzzle, right? You have to be able to get more programs, better programs, moving faster at every part of the pipeline. And that requires you tackling the challenge in this end-to-end systems way. A second benefit of this virtual cycles approach is how it connects the tools, right? It allows us to automatically detect and act on improvements in the places that matter most. So for example, let's say we have a massive improvement in a model or experiment for how we do compound hit ID, in a connected system that lets us quickly detect and then act on a downstream improvement, for example, for validation. And maybe there's also kind of a couple of additional benefits of virtual cycles that are equally important, but not maybe quite as intuitive. So I'm going to take a little bit of time to unpack those. So one big benefit of virtual cycles is that it lets you sample biology and chemistry in this way that's incredibly broad, but also at the same time incredibly deep. So I'm going to use a visual analogy here, where I'm representing the goal of decoding all of biology with the goal of mapping all of the Earth and its topography, all the land and all of the oceans. If you want to do this mapping, there's some places, the flat terrain, right, where you can get away with sampling pretty sparsely, right? If you want to know, you want to be able to kind of detect and then measure the sea level, for example, of all the oceans. You don't have to sample really densely, but you do have to sample really broadly in order to see that 70% of the Earth that is ocean, right? But let's say, on the other hand, that you want to understand the topography on the mountains here in this graph. Now, all of a sudden, you do need to sample quite densely, right? In our analogy here, this mountainous region, it represents the parts of drug discovery that are really complex, right? The places with lots of weird, unexpected activity cliffs. And today, these are exactly the areas of drug discovery that often contains some of the most valuable, interesting, unexpected insights that you could use to start new programs. So the take home here in this analogy is really, if I want to summarize it, that one of the key benefits of virtual cycles is that it allows us to build out an AI strategy aimed at both differentially detecting and densely sample the regions where that's going to matter the most, and at the same time gets a systems understanding of the whole. Here I'm showing some real data at Recursion of the same concept. This is in phenomics. And in the bottom, you see what it looks like when we run experiments in a study, just randomly choosing what experiment to run next, standard approach. And on the top, I'm showing you a virtuous cycle approach, where we're using active learning to look at the experiments that have already been run to make a model-driven decision on what experiments to run next. And the blue line shows you how much incredibly more efficient and fast it is to use this virtuous cycle approach. In fact, in this early study, we got 80% of the benefit running just 40% of the physical experiments. And we believe that this kind of performance that we see in phenomics is something that we can also take to all the different other assays we run across our drug discovery pipeline. Another important benefit of virtual cycles that might not be immediately intuitive is how it scales across experiments. When we run a phenomics experiment here at Recursion, we don't just get the data from that experiment. We're able to compare those results using models with every other experiment we have ever run. So as you see in the chart here, the outcome you get from that is that a linear increase in the cumulative number of experiments that you have run, that's the blue line, that gives you an exponential increase in the data point relationships, that is, in the number of biological insights, a massive flywheel effect. And maybe as a cool aside, make sure you note the y-axis. That is millions of experiments resulting in trillions of biological insights. It's pretty cool. So I'm going to bring it back again to Chris's opening remarks. What underpins this autonomous system, these virtuous cycles? It's really one simple thing. It is the insight that the true key to success in AI, it's not about some specific algorithm, it's not about the transformer architecture or some other specific foundation model, although we do use those as well. Instead, it is the insight that AI-based biology success, that is all about getting increased amounts of compute combined with ever-increasing amounts of broadly usable data in order to create models that solve important, complex problems. Or maybe to say it slightly differently, because this is the take-home of the whole talk, so I'm going to do it one more time. The foundation, the formula for AI success in the modern era, that is the iteration of three things: data, compute, models, as an engine that lets you create ever more valuable products across scientific domains. And for me, there's only one next conclusion from that statement, and that is that the companies that do this well, they are the companies that are going to win. And that's not just an abstract theory, right? We've seen this formula work. So this is the formula that cracked complex games like chess and Go, right? This is the formula that cracked computer vision in the world of images. This is the formula that very recently cracked natural language. And it is the formula that Recursion is using to crack biology. So let's dig into some of the specifics here of these three components, and I'm going to start with compute. About 3 years ago, in 2021, Recursion built our first major supercomputer, BioHive 1. It was one of the top 100. It was the top 100 computer in the world at the time, and it really kicked off a deeper relationship for us with NVIDIA. And as many of you have heard, in 2023, NVIDIA made a $50 million investment in Recursion. And of course, at this same time, there was this massive increase in the world in the appetite, the hunger for compute, broadly, but also, of course, internal to Recursion, so that by the time 2024 came around earlier this year, we stood up BioHive 2, a new generation supercomputer that's fourfold as powerful as BioHive 1, and it was recently ranked as number 35 supercomputer in any industry across the planet. So compute is getting faster at Recursion, but does it matter? You don't have to take my word for it, because our CTO, Ben Mabey, interviewed Joshua Bengio, one of our scientific advisors, about this. He's considered one of the godfathers of deep learning, and this is what Joshua had to say.
We're having a conversation with Joshua Bengio. Do you see any line of sight of the need of compute going down? That was great.
The other way around?
The other way around.
Powerful interview. And I bet you will not be surprised to hear that our in-house data also back up Joshua's laughter and take. So here's an example where I'm showing the improvement of our really critical models in phenomics. And on the x-axis, you see the model parameter number, so the size of the model. And in practice, this is really determined, limited by the amount of compute you have available. And then on the y-axis, you see the performance against a critical biological recall metric. And the basic message here, right, what you see is the more compute, the bigger the model, the more biology you see. And looking at this, there's no indication that we're anywhere close to saturation, right? It's going to keep getting better with more data, more compute, larger models. In fact, spoiler alert, later in this talk, I'm going to show a continuation of this chart, because this right here is just the results from BioHive 1, and we have some hot off the presses from the last few weeks on BioHive 2. But before we get there, we're going to hear from Rory Kelleher, Global Head of Partnering at NVIDIA, who's going to share some words and some thoughts on the intersection of compute and drug discovery into the future. And Rory's actually here today, but I didn't know that, so I made a video.
If you look at like the top computers on the top 500 list, like those are typically national labs, the world of government, like aerospace and engineering. And to have a pharmaceutical company now building a supercomputer that ranks at top 35, I think that's a really important signal. The world of drug discovery is now becoming a technology industry. We've been digitizing biology data at a resolution and at a scale that we haven't actually seen before.
We have this cycle that's really pushing on the flywheel of innovation, from data QC as things are imaged in the lab, to hit discovery, to target optimization. Everything is kind of built off of these high-dimensional embeddings coming from these foundation models. As you add more data and more compute to these trainings, the model gets better.
We can now represent the world of chemistry and biology in a computer. I look at Recursion as like a lighthouse across the industry of this new paradigm of taking large-scale, industrial-scale digitization of biology data and marrying that up with computers.
GPUs and supercomputers in general, they've been in extremely high demand. As the generative AI boom has really taken off, everyone is investing in this area. It's incredibly hard to stand up a new supercluster at this point. We at Recursion, we hear that, but we also like to move at what we call recursion speed. Once we had the supplies, we actually stood up in less than 3 weeks of time.
Oftentimes it's the ones who are willing to take very measured but very bold bets that are going to see some of these outsized returns and capabilities emerge.
Pretty cool. With those words of inspiration, I'm going to transition us back to the pillars of this talk and transition from compute to show you some examples of the virtuous cycles between data generation and models. Put simply, at Recursion, right, we generate data that lets us build models that decode biology, and that gives us insights that we can act on for human health and disease. The first foundational layer of that is to create these highly standardized, high-throughput assays that lets us create massive data sets of high-dimensional, fit for the purpose of machine learning data. And then we build models on top of this data or between these data layers in order to drive our autonomous drug discovery systems. But let's get concrete. Let me show you some very specific examples. So my first example of a data generation modeling loop is for phenomics. This is essentially the unbiased use of microscopy images to study cell morphology, and we use this at Recursion all the way from program initiation through compound lead optimization. On the data generation side of the loop, we have run 250 million of these across dozens of cell types, including really difficult to work with stem cell-derived neurons. And I'll highlight one recent improvement to this assay. We're no longer tied to having to run this in cell paint with fluorescent dyes. We can instead run phenomics in bright field microscopy with multiple time points captured. This is really valuable because it allows us to not just get a single snapshot of what's going on, but to capture the dynamics of how diseases and compounds affect cells over time, right? That's a richer view, a fuller view of what's going on, and it allows us to build new kinds of models. On the modeling side here, I'm showing an example of the power of compute to increase our speed of R&D. So this is the end of the cliffhanger I introduced in the compute section. So this is showing improvements. So on the x-axis here is the model size, and the y-axis is performance on a biological benchmark. So in just 2 weeks of using BioHive 2, our new supercomputer, we were able to generate a new list of candidate models for our in-house production systems. And these new candidate models outperformed our old models in a range of biological metrics. This is valuable, right? Because it represents thousands of biological states, or biological relationships that we were previously blind to, but that we can today use to initiate and drive programs at Recursion. My second example of a data generation modeling loop is transcriptomics. That's the unbiased study of RNA in the cell, and we use this as a complementary, independent data layer next to phenomics. On the data generation side of the loop, we built out a gigantic laboratory for transcriptomics maybe about a year ago, here right across the way, and we've already sequenced over 1 million samples. That's a lot of transcriptomics. And more hot off the presses results, I'm excited to, for the first time, be able to announce that we have built a genome-scale genetic knockout map in transcriptomics in human primary cells. To my knowledge, this is the first demonstration of this kind of data set at this kind of scale ever. Here's just a small, tiny snapshot of that data on the well-known IL-6 studied pathway. So if you look here in the upper right-hand corner, that's showing gene-gene knockout relationships. So red means similarity between two knockout genes, and blue means oppositeness in our high-dimensional model space. And on the lower left-hand corner, you see the same thing, but for phenomics. So one cool outcome here, right, is that transcriptomics recapitulates. It lets us validate the results from phenomics. We see many of the same pathways and relationships, allowing us to use transcriptomics as a validation strategy. But also, there are differences. So here's just an example with STAT3 showing up quite differently within the transcriptomics results. This is expected, right? Transcriptomics captures an upstream signal in the cell from phenomics, which mostly represents the activity of proteins and organelles, right? It's a different time point, and you wouldn't expect it to be all linear in the same relationships. But not only is it expected, it's incredibly valuable, because this means that we have additional real biological signal that we can add into our models to extract even more insight. In fact, on the modeling side here, I'm showing an example of our existing ways of doing multimodal modeling workflows across phenomics and transcriptomics. And I'm showing here that, compared to running either one of these alone, we get a major lift in performance by combining the two. In fact, the numbers here are showing our predictive ability on downstream program-specific assays. That's really valuable because those program-specific assays are often expensive and they're really time-consuming to set up. And you can only use them for one program or a few programs, right? And by replacing those with transcriptomics and phenomics, we've been able to massively increase the scale and throughput in early drug discovery. Another example of a data generation modeling loop at Recursion is ADME. This is essentially the study of what a drug is likely to do in the human body. So for example, blood plasma binding. Is the drug just going to get stuck unintentionally to proteins in the blood? Or microsomal stability. Is the drug likely to just be chewed up by the liver before it has a chance to have its therapeutic effect? And this is something we incorporate in our loops of drug discovery in order to filter out problematic compounds. On the data generation side of the loop, we've been able to combine a number of these assays across human and rat contexts onto a single automated platform, which has resulted in a 90x increase in throughput compared to the standard manual approach. So this is volumes and standardization of data that allows us to create a whole new generation of models. On the modeling side of the loop, I'm showing an example here of the power of these standardized data sets and compute. Maybe I should really quickly explain the y-axis. This is using an external well-known benchmark called the TDC, the Therapeutic Data Commons. It's across 22 widely used ADME assays. And here are the results of a model called MOL-GPS created by Recursion's Valence Labs. It's a graph neural network. It's a chemistry structure-based models that lets you predict properties like the ADME readouts. And it performs really, really well against the TDC benchmark. And you will probably not be surprised at this point in the talk that as you move along the x-axis, the parameter size gets bigger, the model improves a lot. But what is maybe surprising, certainly very good for us, is look what happens when you add in the phenomic data layer to the model. It gets significantly better. In fact, to put a point on it, adding phenomics has the same benefit here as a 50-fold increase in the modeling size. And not only is it an improvement of the new model relative to our old model at Recursion, it also allowed us to beat the state of the art on this TDC benchmark. My next example of a data generation modeling loop is in vivomics. This is ML-enabled animal studies using 24/7 video and animal temperature sensors. And we use this to get an early insight on what a compound is going to do in a whole organism. On the data generation side of the loop, we've been able to scale this to 1,000 data collection cages. And we've also expanded in vivomics so that it can work on rats. That's the second important animal model beyond mice. It's all volume and diversity that helps us build new models. On the modeling side, machine learning lets us take those video and temperature sensors to create drug discovery signal proxy readouts that we can apply across the breadth of Recursion, every program in our portfolio. And one use case that I'm particularly excited about is one where we use this to be able to pick out the right compound from a series at the right dose so that we have the best possible shot on goal when we run complicated downstream efficacy studies. And Laura, in the next talk, is going to show some specific examples of how this works. And my final example of a data generation modeling loop is patient data. That is molecular data like transcriptomics and clinical notes from real people. On the data generation side of this loop, we partner with Tempus to get access to over 20 petabytes of patient oncology data. And with Helix, I think we have a few people from Helix here today, for hundreds and thousands of patient records in a broad range of indications, also far beyond oncology. These data sets are really powerful for us because we're able to combine them directly with our in-house data in transcriptomics and phenomics to build joint multimodal causal models. In fact, on the modeling side, we're in the first 6 months or so now of initiating real programs from these joint causal models, our maps of biology and the patient data. Patient data, right? It's really valuable because it's so relevant, right? It's so rich, but at the same time, it's really hard to work with, right? It's noisy, there's lots of confounding effects, it's incomplete, even with a great data collection from Tempus and Helix, right? So for example, if you want to find a new causal driver in cancer, you're almost always going to be limited by statistical power, right? That effect is going to be really rare, and the data is noisy. And so the value here really is that by combining the patient data with our highly controllable, densely sampled in-house data, we're able to still extract out those targets and be able to validate them in a quick and affordable way on the platform. So to summarize, I've shown you a bunch of examples of the power of focusing on a limited set of high-dimensional data layers. I've also shown you how we apply a whole suite of modern machine learning tools to those layers. And in addition, the kind of extra value, sometimes very surprising value, we get out of combining data layers within one single model. And in the end, I've shown you examples of how by doing this, we're able to get virtuous cycles of both drug discovery insights and ways to improve the system itself. So that at the end of the day, what we have is something much far beyond Tech Bio 1.0. It's not independent point solutions in space, right? It's an interconnected system, end-to-end autonomous automated drug discovery, the vision of the Recursion OS. And so having taken you under the hood of at least some of the pieces of the scientific and technical key components of the OS, I'm going to end this talk by just sharing a couple of highlights of the utility of the outcomes of the OS. And we think of these really as three big, broad categories. One of them, I think, is really well established, the pipeline, and you're going to hear a lot more about this later today. But there are also two newer categories, commercial models and interfaces and tools. Starting with commercial models. We have recently released the first recursion model to the outside world. We call this model Phenom Beta. It was made possible by NVIDIA and their BioNeMo platform. And I can show here on an external set of data that Phenom Beta outperforms what is considered the state-of-the-art, the gold standard in microscopy analysis. In fact, Phenom Beta is a foundation model, so it's built and designed to be able to be used on a broad range of microscopy techniques and other imaging modalities as well. So maybe one day even completely different domains like pathology. It is also designed to be able to be used to answer a really wide range of biological questions, including ones that are just completely different from the things we focus on at Recursion. But I would be remiss to not also mention that, of course, even though it outperforms the external gold standards, it's still not nearly as powerful as the new generation of models that drive internal drug discovery at Recursion. And my second example of utility of the Recursion OS is interfaces and tools. I've shown you lots of tools today in this talk, and I've also shared how at Recursion, these are all connected into an automated joint system. But could we make these tools useful and available for the broader industry? How could they become of value directly also for our partners? How could they be something we share directly with our partners? To solve for this, we built LOW, the LLM orchestrated workflow engine that Chris briefly introduced. In essence, this is a research interface that lets scientists, without any coding ability or kind of technical deep knowledge, be able to access our machine learning models and our experiment orchestration tools. So in essence, that's the LLMs within LOW allowing you to interact with natural language, kind of like you would ask a colleague for help, right? And we're currently in the process of preparing LOW so it can be used by our close colleagues through our partnership with Bayer. Very exciting. You're going to have a chance to see LOW live in action during the demo tours later today, but let me just use an 80s analogies, because I like those, to explain a bit how LOW works. In the early 80s, right, to be able to use a computer, you had to be a real domain expert. You know, it was the field of the chosen few, and the reason for that was that the only way to do it, to get a computer to do something, was through the command line. And then in 1983, the Apple Lisa came out. It was the first computer with a graphical user interface. And all of a sudden, you could make computers do cool things in ways that were way more intuitive and open to the average user. And we believe, and maybe I should kind of put a point on that, right? That was the start, the real start of the personal computing revolution. Right? And we believe that we have the opportunity at Recursion to use LOW to do something conceptually similar, right? That LOW is an opportunity to turn drug discovery into a search problem in a way that's accessible to every scientist. And then the final output of the Recursion OS, of course, is the drug discovery pipeline itself, our internal one and the one with our partners. And Laura is going to come up after me and talk much more about this in depth. So I'm just going to leave you with one parting thought. And that is that this is really the guiding vision of all of the Recursion OS. In fact, it is the guiding vision of everything all of us do here at Recursion. We're not here just to develop the next great drug. We're here to build out and use an industrialized drug discovery system, something that can consistently and repeatedly get the next greatest drug to patients across many disease areas and with ever increasing speed. Thank you. And I guess Ryan is going to do a transition.
Thank you, Lina. I don't know any other investor day that has a Joshua Bengio laugh track, so I hope you all enjoyed that. And now to lead up from our OS platform into our preclinical, we're going to have Laura Schaevitz come up and join us, who is our Senior Vice President and Head of Research. So, Laura, take it away.
Thank you, Ryan. I always find these intros unusual because you intro me and then I intro myself again. So just so everyone, in case you didn't hear it the first time around, I'm Laura Schaevitz. I'm SVP and Head of Research here at Recursion. Now, Lina just talked you through, and Chris before, the power of our massive data sets, our compute and our AI algorithms, and how we put all of those together to massively transform how we are doing drug discovery at research at Recursion today in order to deliver better medicines to patients faster. And so I want to start by talking a little bit about how the Recursion OS is actually changing the shape of the drug discovery funnel. So here you see the traditional drug discovery funnel, where if a traditional pharma company is interested in starting up a new program in a novel area, they are going to have to spin up teams, they might need to find new technologies, they may need new assays, and all of this is going to take them multiple years and many millions of dollars just to start exploring one single novel insight. That's a lot of time and energy and money just for one potential output of our program. At Recursion, we do things very differently. Instead, we use our massive connected data sets to uncover novel biological insights, and then we use these highly automated, industrialized assays that you've heard repeatedly about now in order to quickly validate that those insights are potentially exciting and might be interesting starting points for programs. And so we can do this now instead of millions of dollars in multiple years. We do this in weeks and for tens of thousands of dollars for each of the programs or the insights we're excited about. And so you can see how this massively is transforming the shape of the funnel. We've already explored thousands of insights. We've run hundreds of those through our high throughput and validated assays to move dozens of programs into our preclinical pipeline already. So in addition, because we're running these very standardized assays over and over again, that data all goes back up and helps us continue to improve our predictions. And this is a way for us to also kind of rapidly narrow that funnel. So for example, looking over our last dozens or so of preclinical programs, Lina already kind of stole my thunder here, but we were able to determine that we have a 90% true negative rate. So what that means for me as a drug discoverer is that 90% of our time, if our in silico predictions and our high throughput assays suggest that an insight isn't going to translate into downstream biologically or more relevant biological contexts, 90% of the time, that's exactly what we see. So you can see how that radically transforms the fact that we can now, only with a few tens of thousands of dollars, really make sure that we're working on the programs that are most likely to translate. In addition, because we apply these same principles, we use the same, may actually sound like it starts to get boring over time, but we use these same high throughput assays to answer many of the core questions that we have in drug discovery. That actually also allows us to accelerate the timelines to deliver programs from idea to a candidate molecule. And you'll see that as I talk through a number of examples. So we are radically transforming the shape of the drug discovery funnel from this V to a T shape. And we're doing that so that we can improve the cost, the timelines, and the success rate, because ultimately our goal is to get better medicines to patients faster. So I'm really excited because now I get to take you through 4 case study programs that we have in our internal pipeline right now. So we're going to go pretty quickly through them. But they're great because they highlight what I'm going to show you are early programs, and as they evolve to some of our more later programs, you're going to see how we evolve our process rapidly and efficiently just over the last couple of years. So the first program I want to start with is for Target Epsilon. So this is for an undisclosed indication in fibrosis. And there's high unmet medical need in fibrosis with only a couple of antifibrotics on the market today. And so we are very excited about this program because it represents a novel approach to treating fibrotic disease. This program is also an amazing example of how we can use a single assay in order to answer many of the core questions that we need to ask in a drug discovery program. And so namely, I'm going to show you how we use phenomics generated right here next to us to answer questions, to find hip compounds in order to deconvolute the MOA or the target of those compounds, and how we use phenomics to drive compound optimization, both against the targets that we're interested in, as well as minimizing off-target liabilities. And we can do all of that with a single assay. So this program actually started out as a brute force screen. And for those of you who are unfamiliar with brute force, that simply means that we have to create a disease model, and then we have to put our whole entire library of compounds on top of that disease model to potentially identify hits. But you'll see we actually quickly transitioned this program to using map-based compound optimization. So we didn't stay in that brute force strategy for too long, thankfully. So our goal for this program was to identify compounds that could reverse the fibrotic state that we had induced in human PBMC cells back to a control state. And what you can see on this side of the screen is a number of our hit compounds, and you can see that very clearly we were able to identify compounds that reverted that fibrotic disease back to control at increasing concentrations of those compounds. So in addition to identifying hits, we can also optimize the compounds. So for those of you who are unfamiliar with how our chemists drive compound optimization, they'll take molecules much like the two that you see in the bottom here, and they'll remove some of these like R1, R5, R4 moieties. They'll put some on, they'll take some off, and they'll see what's important for driving either greater potency or potentially reduced potency. And that tells them what parts of the molecule are important. Now the key take home I want you to get from this slide, at the top you're seeing CRC curves, so you can see that when we apply the same compound repeatedly on this assay, we see very consistent results. And if we change out a couple of these moieties, so the R4, if we remove that one or change the R1 to an R2, we see 5-fold reductions in potency. And so this platform enables us to drive potency optimization. And for this program, we were actually able to drive potency optimization over 100-fold in order to get to our clinical candidate, which is now in IND-enabling studies. We can also use phenomics to identify the mechanism of action for these compounds. So what I'm showing you here is a heat map, and let me orient you very quickly. So every row and every column represents a single perturbation, so either a gene knockout or a compound. The darker the red, the more phenomically similar, and so you can see perfect similarity along the diagonal with either compounds to themselves or gene knockouts to themselves. So now if we take 3 hit molecules that we identified in our brute force screen, and we run them now in our broad maps, we can start to identify what's the potential target or mechanism of action for those compounds. And you can see that 2 of our compounds here, REC-A and REC-C, look highly phenosimilar to the knockout of a target gene that we'll call epsilon. And we were able to quickly confirm that these molecules do in fact bind to and inhibit target epsilon. We also found molecules like D, which do not look phenomically similar, and that's denoted by the gray at the bottom, to this initial target. And if we continue to explore the map, we can identify what the potential MOA or target of those compounds are too. And it turns out that that is an MOA that we are uninterested in continuing to optimize against. So here now you're seeing a broader view of our heat maps, and I'm going to highlight here 4 tubulin-related gene knockouts. So you can see they're all phenomically quite similar to one another. And if we look, we see that those compounds which were not phenosimilar to epsilon are in fact highly phenosimilar to these tubulin-related genes. And we were able then to quickly confirm that these compounds are in fact inhibit tubulin polymerization, which is not good. So inhibiting tubulin polymerization basically is cytotoxic for the cell and leads to cell death. So this is an MOA we wanted to stay very far away from. But now, if we go back to those compounds that looked, again, highly phenosimilar to target epsilon in this map, you can see that they're not phenosimilar at all to the tubulin-related genes. So you can see how we can use this single assay to quickly drive compound optimization, both against targets that we're excited about, as well as minimizing these off-target liabilities. And this is very different from how traditional pharma companies would run compound optimization. So in a traditional pharma company, you would have to set up separate assays both to monitor on-target potency improvements, as well as you'd have to know what off-target liabilities you're looking for, and you'd have to separately screen for all of those. And most importantly for them is that that data is only valuable for that one program that they're working on. In contrast, for us, because we can use this one single assay to both drive on-target as well as minimizing off-target liabilities, we can also put that data, and Lina alluded to this, back into our broader data sets. And these, to both improve future predictions as well as, and more importantly, and put this in the back of your mind, these compounds might be interesting starting points for future programs. And so I'll get back to that in a second. But this means we get a lot of value from the fact that we drive, or that we profile our compounds on just this one assay. So now, Lina shared with you our industrialized drug discovery process. Again, this was an early program, and so we only used a subset of the modules for this program, but you can see hopefully how we use a single assay like phenomics to be able to drive on a lot of the core questions that we have in drug discovery. So I'm going to transition now to a second program. And this is our RBM39 program. And I want this to be the beginning of how you see how we continuously evolve and change our process. So we don't run brute force screens anymore. All of the starting points for all of our programs are now through in silico predictions based in our map. And we're excited about this program because this is the first program that was based on an in silico insight that has actually made it to IND enabling studies, and excitingly moving very quickly because we expect to submit the IND in Q3. So this program is based off of the knowledge that a target CDK12 is potentially an interesting therapeutic target and a biomarker for homologous recombination proficient cancers. But this is a really challenging target because of its close homology to CDK13. And we know that inhibiting CDK13 can lead to a number of toxicities. And so our drug discovery scientists took CDK12 and they looked broadly in our whole phenomics map, and they looked for genes that looked highly phenosimilar to CDK12 as potentially an adjacent target to be able to help this patient population. And what they found is RBM39 as one of the most closely phenosimilar genes to CDK12. And actually, so we shared this with many of you actually download day 18 months ago, and it's only in the last couple of months that people have started publishing papers actually sharing more about this finding. So you can see how using our maps too, we're really ahead of the game in finding a lot of these exciting insights. So now if our teams go and they look more broadly in the map, they can also find compounds that look highly phenosimilar to both RBM39 and CDK12. So these might be potentially interesting compounds. But remember, we do not want to generate just another CDK12 inhibitor. So in an ideal world, we'd have an additional data layer that would enable us to predict whether or not these molecules are CDK inhibitors. And we have that in Matchmaker, which we acquired through Cyclica last year. So what I'm showing you, maybe just a second to say what Matchmaker is, since we haven't introduced it yet. It is a method for us to predict protein target ligand binding interactions. And so what I'm showing you here are those predicted binding for 9 different CDKs, and I'm showing you 2 of our hit compounds as well as 2 positive control CDK12 inhibitors. What's important is an in silico binding Z-score of above 2 is considered significant. So you can see in the red box that our 2 hit compounds are not predicted to bind to any CDKs, whereas our 2 positive controls are predicted to bind to multiple CDKs. Now, of course, we can quickly confirm this in an in vitro assay. So what I'm showing you here is a CDK12 activity assay. We see our 2 hit compounds in the pink and the purple. At increasing concentrations, we see very little inhibition of CDK. In converse, we see very significant inhibition in 2 known CDK12 inhibitors. So thereby confirming what our Matchmaker predictions suggested. Now, I won't go through the rest of this program, but what I do want to say about this is how important it is, and you heard both from Lina and Chris, around how we're continuing to improve and build on our predictions. And I think the RBM39 program is a perfect example of exactly how we do that to drive drug discovery more quickly. So what I'm showing you here are the actual numbers of compounds that we profiled through each of a small number of highly standardized assays throughout this process. And you can see where we started in silico with millions of potential options. We were able to quickly narrow down and synthesize only just around 1,000 compounds in phenomics, and so on and so forth, down to that single candidate molecule, which is now in IND-enabling studies. And we did that incredibly quickly, as you can see on the timelines. So going back to our process, for this program, we used a few more of our modules, but actually this program was still fairly early days in our industrialized process. And so our drug discovery team members actually used these modules in a fairly manual way because we were still kind of learning how to use them and how to think about driving drug discovery programs more quickly. But we were able to take the logic of using these modules for this program and really automate and start to create automated decision making. And so the next two programs that I'm going to show you really highlight that once we've got this flywheel spinning, how quickly and efficiently and how many exciting insights we can find using our system. Oh, and not to put too fine a point, we were able to deliver this program in less than half of industry average from a target to an IND. So for these next two case studies, these are very early days, so I'm not going to disclose the targets to you. Hopefully sometime in the near future, I will be able to as we have more data. But again, I think these programs beautifully highlight how when we put the Recursion OS together and the modules together and we automate it all together, how quickly we can move to find exciting insights. So for this program, we had a potentially interesting oncology gene. But what made us really excited about this program was the fact that we identified several hit compounds in our in silico laters that were highly pheno-opposite to this target. And you can see that in the blue in this heat map. And so what that means is that these compounds are predicted to rescue the loss of function or restore the loss of function of this target gene. And because now remember, it only costs us tens of thousands of dollars and weeks to be able to validate these insights versus years in a traditional pharma company, it becomes easy for us to want to immediately generate the data to see whether or not these predictions confirm and whether this might potentially be an exciting program for patients with unmet need. And this program's also really exciting because it's one of the first where we have Tempus insights to support the program as well. And so around the same time we were starting this program, we signed the deal with Tempus and got access to their data sets. And our team quickly, within I think about a month of getting access to the data, had started to build the first causal models. And they independently identified this target as a potentially exciting target for a set of patients with high unmet medical need. And so we're really excited about this program. So now let me walk you through the actual data that we generate. So we have our in silico insight. The next step for us is to use our highly standardized and automated processes for compound selection. And so we have learned over time precisely what compounds we predict will report out positively in downstream assays, and we're constantly reducing those numbers. And then we run them through a couple of our highly standardized and industrialized assays, namely phenomics and transcriptomics. And I'll start with the key takeaway, which was that when we put this compound on our disease model, we see a rescue back to the control healthy state. But diving in maybe just a tiny bit deeper into the data, the red dots that you see on this slide actually represent a variety of different ways that we create our disease state, and the green dots represent a variety of different ways that we create our healthier control cloud. So it's not just a single example. We actually are able to explore a little bit more broadly the potential states around a particular disease. And again, when we put this compound on top of the gene knockout, we're seeing a reversal of both the morphology as read out by phenomics as well as the transcriptomics profiles back towards our healthy state. And for me as the head of research, what I'm particularly excited about with this is that our teams, we love you all, don't actually look at any of this data. So these experiments are run, this data is generated, and they are analyzed. They only look at the data or in programs like this where we see positive data from both our phenomics and transcriptomics assays. Those are the only experiments that our teams are looking at. And for this program, we take a deeper look at this molecule, and we realize that based off of the phenomics EC50 and PK profiles, that we actually believed that we could get sufficient exposures to go directly into an in vivo animal study. And so that is what we did. But on the way, Lina told you how we always run in vivomics digital tolerability studies. So let me show you that data. So you can see on the far side of the screen just one of the readouts from our digital tolerability studies. So we run tolerability in order to be able to quickly identify what is the maximum dose that we think an animal can tolerate over a much longer efficacy study. And that allows us to always pick the right doses for efficacy studies. So what you can see over the course of the 7 days of dosing here, it's a little challenging to see, but in the black are the vehicle animals, and we're reading out motion here, so you can see they're far more active at night than they are during the day. We see similar results for both the 30 mg per kg dose group in blue, the 100 in red, but once you get to the highest 300 mg per kg dose, if you look closely, you can actually see that those animals are not nearly as active. And this is just one of the measures that we get on digital tolerability that told us that this 300 mg per kg dose is unlikely to be tolerated during a longer efficacy study. So we took that 100 mg per kg dose as well as a couple of lower doses, and we put them into an in vivo efficacy model with tumors with a mutation of interest. And I want to remind you, this is an unoptimized molecule that we pulled directly out of our maps. We already see a 66% tumor growth inhibition with this molecule. So we're pretty psyched about this program. And our teams are working right now to continue to optimize the molecule using generative AI and some of our digital chemistry tools, and again, continuing to use phenomics and our ADME platforms to, inside joke, in order to be able to quickly optimize this molecule. And we've already seen some, even just over the last couple of weeks, improvements in exposure. So hopefully more data to come on this program soon. So this is the first program now we're putting together end-to-end all of the modules and adding in some of the automated decision making. And so you can see for this program, when we do that, we were able to go from an idea in the map to delivering a lead nomination program in just 10 months, and that includes the in vivo efficacy data. So that's very fast. Your traditional pharma company is still probably spinning up assays and teams at this point, and we have a brand new program that we're really excited about. So for the last program, you're going to see a lot of this. I'm not going to take you through all of the data. I'm going to focus for this program on the fact that we found a compound in our maps for what was previously considered to be an undruggable target. So remember back when I talked about Epsilon, how I talked about the value of using a small set of standardized assays is that all of those compounds go back up into the top of the funnel as potential future starting points for future programs. Well, that is exactly what happened for this program. So the lead compound for this was actually a compound we were testing in a completely different program, I don't know, maybe a year and a half before. But we found it as a strong hit for this oncology target that had been considered undruggable. So let me show you quickly the heat map. Here you can see the knockout of the target gene, and you can see our compound of interest looks highly phenomically similar to that target gene. Now at this point, what we don't know is whether or not the compound's acting in an adjacent pathway, somewhere upstream or downstream of that target, or whether it's actually physically binding to that protein. But we do know that it looks, that compound phenomically looks similar to the knockout of the target. But let me skip straight to the most important slide here. We had other reasons to believe that it was quite possible that this compound was in fact a direct binder of that target. And so we went straight to generating this data in SETSA. Let me orient you a bit to this. So RecJ is the compound that was our hit that looks highly phenomically similar to our target. And K is a close analog that actually has no phenosimilarity to our target. And what you can see here is at increasing concentrations, we see the amount of protein of our target stabilized at increasing concentrations, which tells us that this compound is a direct binder of this target protein. So very exciting that we were able to find, through in silico prediction, a new compound to bind what was previously considered undruggable. Now, we used the same process as I showed you in the last case study or program to drive this program relatively quickly. I don't have time to show you all of the data, but please know we took that unoptimized molecule and we put it into, again, it looked like it had the appropriate exposure, so we put it directly into an in vivo animal model, and we saw tumor growth inhibition in that model as well. And we were able to run this program now end to end in 8 months. So you can see how our flywheel is enabling us to move faster and faster to deliver more and more exciting programs over time. So I'm going to finish by sharing a little bit more about how we continue to improve and find the really exciting biological insights that we want to put into the top of our funnel. So I think Lina talked about the trillions of relationships. I think her graph only went up to 7 trillion, but I'm sure at this point it's got to be higher than 7 trillion relationships. So this is really challenging if we wanted to do, I should say we can't do this in a manual process, right? To explore all of those relationships. And so this is where we have started to build large language models in order for us to be able to identify what are the most exciting insights that we want to use as starting points for future programs. And so what I've graphed here in every dot is a potential target for the beginning of a program. And we've used large language models in order to plot these both on how much is known in the public data sets on the vertical axis, as well as how much recursion arbitrage do we have for that target being potentially interesting and exciting for oncology. And this is a way for us to much more rapidly identify what are the novel insights that Recursion has. Remember, we're not interested in developing just the next drug that everybody else has. Where are our Recursion novel insights that we could use as starting points for programs? In the blue, this is a great confirmation that how we've tuned our large language models is really working. In this quadrant, you see genes where there's a lot of data in the public domain, as well as we have a lot of data within our Recursion proprietary data to suggest that these genes are highly relevant to oncology. And we've highlighted just a couple here, but you'll see typical targets like EGFR, BRAF. So this is telling us that we have built, or we've tuned our large language models in an effective way. The purple is where we're really excited about spending the majority of our time. So this is the area where there's very little information on these genes being associated with oncology in the public data sets, but where we have some pretty exciting insights within our Recursion proprietary data sets to suggest that these might be exciting starting points for oncology. So we're really focused here, and we are putting these back up into the top of our funnel, and we're using those to drive our newest, continuing to drive our newest programs, and we already have a number in our hit to lead stage as well as our lead op stage. So let me leave you with 3 key takeaways. At Recursion, we do drug discovery differently than the industry. Instead of spending millions of dollars spinning up teams in multiple years, we've turned drug discovery and identification of novel biological targets into a massive search problem. Second, we use a small set of highly standardized and industrialized assays to drive and answer many of the core questions that are essential in to drive a program from an idea to a candidate molecule in drug discovery. And by doing this, we not only generate the data for our own programs, we generate data that enables us to continue to improve our predictions and as potential starting points for future programs. And the last is we are changing the shape of the drug discovery funnel. By employing our Recursion OS, tying it together end to end, automating our decision-making processes as we learn, we are transforming what was once considered a V-shaped drug discovery funnel into a T-shaped drug discovery funnel, which is going to allow us to deliver better medicines to patients faster.
Thank you so much, Laura. We're going to mix things up a little bit here. So this is the last section before we go on tours and demos, and you get to see everything, all the fun stuff. But I'm excited to welcome up Dr. David Mauro, who's our Chief Medical Officer, and Dr. Deepak Nijhawan, the Distinguished Chair in Biomedical Science for UT Southwestern. So, gentlemen, have a seat.
Testing. Excellent, good job. I've known Deepak now for 6 months, and he is a rather humble man, so indulge me while I talk to you a little bit about who he is and his background. So Deepak is a MD PhD by training, trained in medicine at the Mass General, did his fellowship at the Farber and spent some time at the Broad. Since that time, he moved to UT Southwestern, where he is associate professor in the Department of Medicine, also a member of the oncology division, and he is a UT Southwestern presidential scholar, as well as Ryan mentioned, distinguished chair, the Joseph Sandberg Chair. In other words, he has the type of academic career that I was not smart enough to have. So welcome to Deepak. I want to start at sort of a local level and then broaden out. And I wanted to, you know a little bit about our RBM39 program. When I came to Recursion about a year ago and I started to learn about the program, I reached out to my network and said, I'd like to really sort of bounce this off of a real KOL. And person after person said, you really have to talk to Deepak. Deepak is sort of the father of RBM39. But I guess sort of the first question that I'll ask, about our program. Could you talk to myself and the audience a bit about sort of the history of this target from in your lab and sort of where it's been and where it's going?
Sure. Well, first of all, I want to thank you for having me. It's really a treat to hear about this program and about this tremendous platform for phenotypic discovery. So maybe just by way of introduction, I can talk about, a little bit on how we got into RBM39. We never set out to study that. And our program is in some ways similar to what you guys are doing, but it's a single, I'm a single person. We don't have a giant platform. So, I trained as an internist and then as an oncologist, and I was a biochemist by training, and then I landed at the Broad Institute. And I wanted to go back. When I was at the Broad Institute, there was, kind of early days of RNA interference and nominating targets. I would watch people kind of get a list of targets, and they all had assigned some statistical value, and they would just kind of go down the list and look for which ones were druggable. So they'd be down to 10, 15, 20, looking for a kinase and looking for what's druggable. Because at the time, we had all these proteins that could be potential targets in disease, but no way to really target them with small molecules. And so I moved to Southwestern, actually, and Steve McKnight recruited me there, and he had really built a department around phenotypic discovery. And the idea was to start with a small molecule and then understand its target, and then by definition, it would be druggable. And so Steve really convinced me to build an entire program based solely on that. So we started to use kind of newer technologies that had come about, like click chemistry and forward genetics, to take small molecules, kind of orphan compounds, and try to understand how they were actually working. Because small molecules may be 400 or 500 Daltons, and as Laura just showed, it can land in all these different places in the cell, depending on concentration, into little pockets of proteins and affect function in a really precise way, some that you want on target and some that you don't. And so the challenge is kind of like a needle in a haystack. Can you take this small molecule where you have no idea how it works, you know that it's killing a cancer cell, and can you find out how that happens? And so what we've done over the years is to build genetic tools to basically give us a single mutation that takes us right to the key binding site of that molecule. And so that's what happened with this program. We built our first generation forward genetic system, and we started to put a lot of orphan compounds through. One of those compounds was this compound that was discovered by Eisai Pharmaceuticals back in the late 90s. And that compound, we found mutations in RBM39 led to resistance. And as it turns out, this compound turned out to be a molecular glue, and RBM39 was recruited to an E3 ubiquitin ligase. So, we did really kind of deductive reductionist science to get to the mechanism for how that chemical targeted RBM39, which I'll say is, RBM39 would be a conventionally undruggable protein. And nobody, unless you're approaching it phenotypically, no team of medicinal chemists or structural biologists could imagine how you would get a small molecule to target RBM39. So that's one of the real benefits of phenotypic discovery with small molecules is you end up, finding things that people couldn't predict.
So if you look at the literature, RBM39 has been a target that's been around for a while. You discovered it, you in your lab. It's been brought into the clinic in a relatively narrow indication. It's been evaluated in AML and MDS. Could you talk about some of the learnings, and maybe the studies were not that positive, right? It clearly demonstrated target engagement, but it has not lived up to the expectations of when it came into the clinic as a very exciting target. Any thoughts? I mean, it's a complicated question, right? But any thoughts in terms of some of the, I don't want to say missteps, but the lack of prediction preclinically to what was seen in the clinic, and then I'll sort of talk about our approach.
Well, I think, drug discovery is a checklist. You have to have all these various things checked off in order to have a positive result in the clinic. You have to know who to treat. You need to have the right small molecule. It needs to be bioavailable. And that's why you end up getting these kind of screening funnels. And I think Recursion is kind of turning that upside down. So it's not a funnel necessarily anymore. So in this case, this molecule was discovered and these trials were run in the late 90s, early 2000s without knowledge of the target. So if you don't know the mechanism, you have a lot of risk. You don't know if your compound's bioavailable. You don't have pharmacodynamic markers. You don't know if it's, like someone said, all bound to protein. Is it actually getting to the target? So I think this was, and most importantly, you don't know who to treat. I think, over the last 25 years, it's widely appreciated now, and we know this in the clinic, that, no one's cancer is alike, that lung cancer, any cancer, is a whole different set of diseases. And so you need to know exactly which patients are going to depend on a particular target. And if you imagine you have no idea, you're putting this molecule in a person and you don't really even know what you're targeting.
I mean, one of the things that struck me when I came to the company is, have we really uncovered novel biology around RBM39? Clearly there are indications that one could go after with a better molecule, and we believe we have a better molecule, but that's not what Recursion is trying to do, right? We're really trying to utilize our arbitrage to go after unique biology. Now, a lot of that we've talked about, but could you sort of comment on not only the approach, but some of the data that you've seen and we've talked about in the HR proficient solid tumors?
Well, I think that's a novel insight that your group has come up with. I think it's interesting. I just sent you this manuscript recently where people had done phenotypic screens now with RBM39 degraders in PDX models, across multiple PDX models, and came to the same conclusion that your algorithms predicted. And so I do think that is the key element for that, is that, once you have, let's say, a small molecule targeting this now undruggable protein, is there a clinical problem that you can solve with that, and who are those people?
So let's pan out a bit. We've talked a bit about RBM39. I've looked at your work over the years, and your lab has evolved, right? And I think I would characterize Deepak's lab as somewhat atypical in the world of academic medicine. Could you just sort of talk to the audience a bit about how your lab has evolved and why it has evolved in terms of using a phenotypic approach or a phenomics-based approach? And perhaps the second part of the question is, why is it a novel approach? Why aren't more labs investing time and effort and thought into this sort of approach? So kind of
that's a question I think about a lot, because actually phenotypic drug discovery is how drugs used to be discovered, how many of the drugs we use in the clinic were discovered. And in the last 25 years, it's really switched to a target-based approach. And that was for some of the reasons that the RBM39 degraders failed. We didn't know the target. Without knowing the target, it was really difficult to advance drugs that were discovered phenotypically. And as a result, it became expensive and uncertain. And now you can go with a target-based approach, and you have a precise protein, you know what protein you want to target, but you have an entirely new challenge, is how do you get a small molecule to target that protein? And as a consequence of that, the number of targets is actually really finite. So you have an intense amount of kind of competition on a small number of targets, and there's this vast amount of biologic space that's kind of unexplored. And I think phenotypic screening can now come back because things can be done in parallel. So you can look for compounds and targets and patients all at the same time. Whereas in the past, some of the most successful drugs, like lenalidomide, for instance, we were talking about, the phenotype was discovered in the clinic, 15 years into the process, and turns out to be a blockbuster drug, but that's a long time and a lot of uncertainty. So with the tools that you guys have and the tools that have come now, you can find the molecules, the targets, and potentially the patients much quicker, and start to explore this space. So we're not targeting the same proteins, over and over again. And I think the models for different companies have become kind of very risk averse, so that people are targeting the androgen receptor again in a slightly better way, and that can become, I think we're leaving a lot of potential clinical opportunities on the table by focusing on just a small number of targets.
One of the things, I've worked on probably 20, 30 different molecules in the clinic, and I've never worked on a molecule that did not demonstrate some degree of activity. But the challenge has always been trying to identify why patient X responded and patient Y responded. Right? So it's my bias that it is as much about the target as equally to understanding the patient. And I wonder if you can sort of talk about the prioritization in your mind in the characteristics of what molecules should have when they come into clinic, and talk about sort of, I'll let you chat about that.
Well, I think, going back to patient selection again, how early in your clinical development pipeline can you see responses? How well can you predict who would respond? All of these things will really, as you, of course, as all of you know, as you move along in the process, it becomes much more expensive. The experiments are much more expensive, the trials. So I think having, and in addition to that, I think kind of pharmacodynamic markers. When the first RBM39 degraders in the 2000s were used, they were dosed at dose-limiting toxicities. So, having knowledge of the mechanism, and I think your pipeline here, if I could comment on it, can really nominate new targets. And then if you add on to that really rigorous kind of mechanistic mechanism of action and pharmacodynamic markers, you're poised to really explore this space.
So talk to us a little bit about where you think we'll be in a few years from now. Do you expect this approach to have greater uptake? Do you still envision Recursion as sort of leading the field, but a very small field? Talk to us about sort of not only in your view across other industry, across companies, but even talk about sort of your view academically. Do you see more folks, more labs embracing this approach, or do you think that it's simply going to be a matter of time where people will just follow, the leader?
Well, as you mentioned, my academic approach is really unique. Most academics focus on one disease or one problem, and we've decided to focus on, there's thousands of chemicals that kill cancer cells. Let's just find out how they all work. And once we know how they all work, we'll have a much larger toolkit for potentially treating disease. And I think that the academic model doesn't favor that kind of research, but I do think Chris showed this graph of the loss of efficiency. So how can we have so much technological advancement in chemistry, structure, cryo-EM, and still lose efficiency? And I think it's because we're way too focused on a narrow list of targets. And so I think Recursion's efforts to expand target space and to take those to the clinic quickly will potentially kind of turn the tide. I think academics oftentimes will follow, unfortunately, because of the academic model. When recursion is successful, phenotypic screening will be back in favor academically too.
Because as you mentioned, right, it was right years ago, right. Phenotypic screening was the way to go, and you sort of talked about the evolution. Do you think that there's the sort of lack of embrace is an unfamiliarity with the technology? I know that, look, a lot of when I speak to investigators about recursion, and I talk about our approaches, oftentimes, and these are very, very bright people, their eyes glaze over, right? Just talk to me about the target, talk to me about the population, talk to me about the trial. So do you think it's a bit of lack of understanding, which could be
maybe also talk about who else is targeting that protein?
Right, yeah, right, the other piece, right. So is it, again, I'm trying to, because this is important. There, a lot of people here, some of which have embraced technology and what we're doing, and others here that are appropriately skeptical. So, talk to us a little bit about the skepticism. And is it really going to be simply a matter of time between validating targets in the clinic, or do you think it's going to be something more than that?
Well, I think it's going to be about the beginnings of some clinical success following targets that are not conventional. I also think that right now you have KRAS as a target, clearly mutated, new compounds that hit there, and then you have a whole bunch of companies that are in kind of a fast follow, trying to get the better KRAS inhibitor, or we saw it with CDK4, CDK6 inhibitors. So I think Recursion has a challenge of kind of being the pioneer. The very first ones to describe all of your programs are likely first-in-class targets. And so I think I don't think right now there's been appetite in big pharma or small biotech to really have the courage to pursue these kind of phenotypic studies. So I think once you demonstrate their success, one, you're going to have a lot of fast follow, which I'm sure you're ready for. And two, I think you will open up that phenotypic screening might be actually more productive, more profitable, because you're in a novel target space and you're ahead of everyone.
So I'll finish up on this. Deepak is here on double duty. He flew back from Scotland to participate in Download Day, and he's also meeting with our discovery team later today. We'll hear about partnerships that we are doing with companies. I just want to talk to you a bit about potential partnerships with Recursion and academic labs. You're here. We're doing work together. Where do you see the opportunity? Sort of the complementary approach. What can you help us do, or any guidance that you can provide?
Yeah, I mean, for us, we have our own mini platform, where we take a small molecule where we have no idea where it goes in the cell to kill a cancer cell, and then our platform generates a result where it says it's going exactly to this place. And that mutation and that knowledge provides, a tremendous asset to lead optimization and drug development, because it helps you pinpoint exactly what's an on-target effect. And so that's some place where we could help with compounds that you have, where you have some promising phenotypes, phenomics, but you don't know the exact mechanism. Because even though you want to move quickly, with just 3,000 or 4,000 compounds, quickly get to IND, you don't want to miss anything. The stakes are high. So all the knowledge you can have, cryo-EM, in vitro assays, all of the things that that come with traditional target-based programs, you also can deploy with knowing the mechanism. That's one place we can help, and I would say that's the best place. The other ways is that for chemicals out there, maybe even in the public space or in our space, knowing who to treat, who would benefit.
So again, I want to thank Deepak for coming, spending time. I think he arrived late last night around 11 o'clock, but he looks fresh and bright-eyed. And thank you for his partnership and his guidance, and really thanks for the chat.
Thank you, David.
Thank you, David. Thank you, Deepak. Okay, we're going to get started. I think we have critical mass at this point. We have a very exciting afternoon for you all, and I'm excited to introduce the folks that are going to kick it off. I'm going to start with our newest board member and soon-to-be Chief R&D and Commercial Officer, Nijhawan. So, Nijhawan, if you could come up and join us.
Hello, everyone. I get the afternoon post-lunch slot, so I'll bring the energy here. Thank you, everyone, for making the time. It's really, really good to be here. What a morning. I've been following Recursion for a while. I'll tell you a little bit about me. But it was just great to see the constant evolution that's been happening from the presentations from Chris, Lina, Laura, and much more to come. But given this is the first time we're meeting, I thought I'd spend a little bit of time telling you a bit about myself. So I've been at J&J, and I'm joining Recursion from J&J for 6 years. At J&J, I headed up the pharma portfolio across oncology, immunology, neuroscience, and more, as well as being their chief data science officer. So I built up the data science team from scratch there. So you can imagine, and you'll hear this quite a bit when I'm speaking, the convergence of the two disciplines, pharma, making medicines, and leveraging AI and technology to do it in a better way is something that's going to be consistent in many things that I say today. My background prior to that, PhD in organic chemistry. Yes, I love pain. I also, when it was not cool, a background in computer science and a lot of business background as well. And I think the convergence of the three is something you're hearing a lot in the conversations today, which is it's important not to just to have good science and technology, but to actually make sure it's outcomes focused. So that pragmatic and disciplined approach, I think, is going to be really important from what I see in the space. So with that preamble, I'm going to focus the next few minutes, on 3 questions that I get way too often. First question, why AI in healthcare? Although I have to say I'm getting that question less. So I'm going to spend a little bit of time. The second question is, what can AI do in the life sciences space? What's real? What's not? What is it going to take to get us to the next level? And then the third question that I've been getting a lot in the last month and a half is, why Recursion? So I'll tell you a little bit about why, and also what are the areas I think we need to do more, because it's a constant evolution to be the best. So with that, let's get going. So as you'll get to know me a little bit, you'll see all my presentations will start and end with the patient in mind. I just got back from a trip in Singapore where I was with my mom for a few weeks. Really, really frustrating time. She has Parkinson's and now dementia. And you just get the runaround from physicians because no one really understands the biology of why you get dementia. And quite frankly, it's not even one disease. So how are you going to treat something when you don't even understand the first order principles well? And when people ask me, why do we need AI in healthcare? The answer is simple, right? Look at all the patients that are waiting. And on top of that, you can add stats to that, right? 10% of the drugs fail. 10% of the drugs succeed, 90% of the drugs fail. But if you go a little bit deeper, across the healthcare space, there are many areas where you can actually have impact, and we're starting to see that using AI. And a lot of you know this, right? But I think the one part that gets not the attention that it needs is really around better understanding disease causality. Again, if you don't understand what problem you're solving for, how are you going to do it in a better way? Now, there has been progress with medicines and genetics. Some of you saw Alnylam's result, very good to see with RNA therapeutics, but there's so much more to be done. And if you can understand that leveraging multimodal data, then you get to a point where you can actually diagnose disease earlier. I mean, for today, 40 to 50% of cancers get diagnosed too late. So no matter what drug you have, the outcomes are impacted because you're not getting to the patients early enough. And then the next two areas where I'm going to spend more time on is really around developing molecules. Let's think about this for a second. The chemical space, if you're making small molecules, that's 10 to the 60th. I'm a beach person, and I've heard this analogy before, so it's not mine, not an original one. But there are more molecules, and the chemical space is bigger than the number of grains on a beach in the entire world. So that is a big problem to solve for. And that's where you need AI, you need compute, you need scalability, and so forth. Same thing in biology, trillions of cells. You need to understand that better using AI, using compute. But it's not just enough to actually make a good molecule, as many of you know. Therapeutic development is a huge area where a lot of molecules fail. And to me, that's almost the last mile in terms of ensuring that we're actually designing the programs the right way, that the molecules have the right PK/PD. You heard a little bit about some of the algorithms. Supremely important to do that better. And then the super last mile, which is actually ensuring the medicines get to patients. This is where personalization, access, and adoption is huge. And last but not the least, we talk a lot about productivity. It just makes sense to do more with less and then improve the quality of care by doing so. But let's zoom in a little bit more in terms of the life sciences space, which is why we are here today. So we're going from a world of, again, as I mentioned, genomics, there's been some great strides, RNA therapeutics, some MD simulations that have cracked the code for some hard molecules, but it's still not enough. And then if you look in development, and this goes back to the example I used earlier, the imprecise disease definitions, how many diseases today are named after either the physician that identified the disease, or it's just a broad description of the disease. It's not based on the biology that's driving the disease. So it's really hard to get to the right patient selection if you don't understand what's causing the problem. So look, there's a lot to be done here, and I'll go through it more in detail, but multi-omics data is creating a massive difference in better understanding not just the target, but if you jump into development, being able to stratify your patients better so that you can actually realize the promise of precision medicine. And why do you do it? You want to take the V, which Laura mentioned, and you want to turn it into a T. So better predictions up front, pull that through in development, and ensure that you do it better, faster. Sounds easy, not really. I mean, if there's something that's humbling in my entire career has been making medicines. It's really complicated. So then let's get to the second question that I get all the time. Where are we today with AI? A lot of you have seen the S-curve. We're not all the way at the top. We're not all the way at the bottom. We're at a really interesting point. I would call it the pre-tipping point. I don't think we're at the tipping point yet. And the reason for that is the following. Some of the examples here, just listed on the left-hand side, are great areas of progress. Like AlphaFold, I mean, as a chemist who was actually also leveraging computer science to be able to predict the protein folding and then find something that would bind and ensure that that doesn't get chewed up immediately before you even do any experiments, it's close to my heart. Huge amount of progress made there. Also, in terms of how do we better execute our trials, ensuring that there's greater diversity. Some of the prior work that I did was focused on that, seeing upwards of 2 to 3x increase in enrollment rates, getting diverse patients faster because you are actually going to where the patients are at. A lot of publications, of course, from the FDA in terms of guidance as to how do we actually leverage AI in drug discovery and development. All that's good, and also some great examples from the work that Recursion has done, looking at molecules, this is during COVID, that exist in the real world, and using phenomics to be able to anticipate which ones could be effective. But here's one problem. You heard themes of this. A lot of these are really important, specific point solutions. But in order to actually make a molecule and then turn it into a medicine and get it to the patient, there are multiple steps involved. Can't fall in love with an algorithm. Fall in love with the outcome, which for us is to make medicines for patients. Next point, which I think is extremely important, and I've seen this be challenging sometimes, which is then is the exuberance of, we need to fix it all. The cost to change and pioneer in this space is high. So to prioritize where you focus, where you innovate, where it matters the most, it's extremely important to be smart about it, and I'll talk more about that. The third, the amount, I think somebody mentioned at the Fireside Chat that whether it's pharma and whether it's biotech, the adoption hasn't been what it needs to be. And one of the big reasons for that is some of these solutions are not as democratized yet. It's true. But the other is culture and agility. Because if you don't know what good looks like in your organization, it's really hard to cut through the hype curve. And I think that's one area which I call it the bilingual talent and culture. Is crucial to the companies that not only you want to work on the right problems, you want to prioritize where to innovate, you want to have the agility to stay ahead of the curve, but you also want to ensure that you have people that will actually evolve with this space and not have silos, which is something we see so often in this industry. It's a team sport, as they say. So I'm going to share with you my cheat sheet that I've had for some time, constantly evolves because I got to evolve too, of what I think the pharma of tomorrow will look like. And then I'll talk about where does Recursion fit in that? So you won't be surprised to see. Pipeline, whether it's an internal or external pipeline with partnerships, more comprehensive understanding of the biology, supremely important. You can't not do that. This is one of the biggest reasons why we have such high failure rates. Second, novel molecules, but I want to put an asterisk on this. A lot of people are designing molecules that look really cool, but you actually have to have ones that have the right PK/PD profile so that you don't put it in the clinic and then it fails. This happens a lot because people rush things to the clinic. So quality, and that's something that's always been a focus, quality is going to be paramount, more important than speed and cost. Precision development, oh my gosh, how long have we talked about precision medicine? I feel like forever. But if you understand the biology better, you can actually do precision medicine better. And trial design and execution, sometimes people think of it as an afterthought, but 70% of the dollars to make a drug actually goes in development, cost and time. As important as the output is the how, which is the foundation. So I have a bunch here. But if you look at a pharma today, the wet lab is there, the dry lab isn't. Cutting edge AI, machine learning, a few do, slightly biased. High quality data at scale, proprietary data. I think Chris brought up this point that, commercially available data, you hit a limit at some point. You hit a limit. And the reality is, most of the biology, we understand scattered little aspects of it. You need to connect it, because we are a connected system, right? You can't just solve a problem in a specific point. So that is something that doesn't exist. And this is one of the reasons why on that S curve, sometimes people will ask me, there's been so much investment. Why isn't this point higher up in the curve? Because in order to reach that tipping point, you need a huge amount of investment to get the data right? To do the right experiments, to make sure that it's actually generalizable. It's not easy to do. And of course, the little thing around world-class compute. But I will say that the pharma of tomorrow will have many elements that are different, but the one thing that's going to remain the same is to actually make transformational medicines. That word transformational, underlined, because access, adoption, pricing, I didn't talk about the commercialization part, is getting harder and harder, and the bar is getting higher. So now, where does Recursion fit in that today? So I'll share with you a little bit of my cheat sheet on that. Look, Recursion started in the drug discovery space. Makes sense. Huge focus on phenomics, and then building on that with transcriptomic. I really like the examples of triangulating and being able to validate the dry and the wet lab. That's really great to see. Digital in vivo, I call it the digital endpoints for animals. Look, if you have more sensitivity in your animal models, but there's much more that can be done with organoids and other human models as well, which I know there's thinking around that. Chris and I have known each other for some time. And I remember even 18 months ago, if you looked at this map in my cheat sheet for Recursion, it was very dark in the therapeutic hypothesis space and some of the foundations. But very quickly, the company has evolved from molecular invention, I call it generation 1.0, which is repurposed molecules, to molecules that are made in Recursion. That is extremely important, because one thing that I've seen here, and I've been pleased to see, is the maniacal focus on actually making medicines, creating drugs. But you can only do it so well unless you develop the most important value asset in any pharma, tech, bio, we can call it whatever we want, company, which is the molecule. The other aspect of it that I think is important, and the team touched on it, is data and AI software. How do you actually democratize this new way of drug discovery across the industry, right? And then more and more work that's being done, and I think this is an area that I'll definitely be leaning into, is how do we go deeper in the discovery space, and then also how do we ensure the emerging work that's being done, leveraging these innovative approaches in development, are scaled. But let me give you another page of my cheat sheet. So as I've been having conversations with various folks in Recursion, I think about some emerging proof points, because that's what makes things compelling for me. I like specificity. So we talk about novel biology that nobody has done before. You heard some of the examples from RBM39, but there's over a dozen examples that Laura and the team are working on in the early and late discovery space. For the size of the team, and if you look at industry standards, that's quite a lot. So efficiency is something that you're starting to see. And it's not just for the internal pipeline. It's also for the external partnerships with Roche and Bayer, which I'm sure Matt can touch on more. But it's not just good examples in novel biology in the therapeutic hypothesis space, but also making molecules. All of these programs that I just mentioned, over a dozen, they have molecules that have been made in Recursion. I think that's really important, and we'll talk a little bit about how. But 60% of them use Gen AI. I know a lot of you will probably ask me, why isn't it 100%? Because you use technology where you need it, not just because you have to, right? And then the other aspect that was quite compelling to me is, I think it was over the last 4 or 5 years, looking at the speed and the rate being 3x faster, 2.5 times lower, and elevating the quality of the programs. That's where you cannot do that without industrialized workflows. You cannot do that with the scale. You cannot do that without the proprietary data that's been built up over the last few years. Precision development, 7 programs, 5 in the clinic. And I'll talk a little bit about one of the examples where the team is actually focused on doing recruitment in a very different way, orders of magnitude faster than you could do traditionally. But in order to do all of this, and I get this question a lot too, is there going to be one algorithm that can take care of everything? Not yet. But it's important to stitch together multiple aspects that's needed, that's really important to get to a drug. So Phenom 1 around phenomics. What I really like is actually on the chemistry front, having different algorithms for hit ID, for ADME, which I can't emphasize enough the need for that, but also for lead optimization. Lead optimization is probably one of the biggest things that the industry gets hung up on. Super important to do that better. And then Low, which actually integrates, and I'm sure some of you saw it over the demos, integrates a lot of these workflows. So if you look at a lot of pharma companies even today, the data is being generated, not in as a systematic way. Many algorithms are being developed, but it's really hard to bring it all together and to make sure that scientists are actually using it in a systematic approach. I won't go through the rest in terms of compute, but I'm just going to click through some of these examples. So RBM39, and you heard a lot about it, yes, the biology, it's not just that the biology is new, but having a deeper understanding of patient selection is going to set you up for success in the clinic. But I want to reinforce something that Laura said. The 18 months is excellent. But if you look at the number of molecules that were made, I think you said under 1,000, right, 955. I mean, in industry generally, you'd make thousands of molecules. So the waste that goes through it, because you're searching, right? You're searching, and it's not the most efficient way of searching. I think that's something to emphasize. That's where you see the efficiency gain. And then you've seen some of these stats before, but again, I mean, if I look at the size of the teams that are working on this, it's a fraction of what you see in industry. So I think that is important to emphasize. You know, we talk about scale, we talk about efficiency, we talk about having better probability of success. All of that investment is what's needed to actually get it there. And now we need to focus on the quality of the molecules to ensure what gets in the clinic does really well. 2 more examples for me. I really like this one. First, because having been someone in the pharma industry, hundreds of programs, so many programs get delayed, and seeing how you can change that, it's not just impressive to see that you're doing it from 4 to 6 months, and this is for the Axon-1 program, APC, and you can recruit that cohort, and the team was able to in 4 to 6 weeks. But if you just look at the image, today we usually start the approaches. You start by asking the sites, how many patients do you have that fit this profile? And then you go back and forth and you do a little dance, and I've done the analysis, the success rate, like the realization of that is very low. Anyways, and you do all that and then finally get the patients. But in this case, in this approach where you actually use genomics data, real world data, AI, to actually figure out in this country, where can I find in an anonymized, de-identified way, where can I find the eligible patient populations, where do they exist? And then go recruit them. It's much faster, it's much smarter, and quite frankly, it is the heart of patient centricity, which we've talked about for so long. So watch this to be an area that gets scaled. All right. And look, to do all of this, and I know a version of the slide was shown before, but as I was having conversations with folks, for me, my brain works in discovery, development, commercialization, so I had to map it. And I think it's very important to see here that it's not just building modules and algorithms for the sake of it, but it's actually building the ones that matter. Because if you don't have phenomics, then you're not able to predict some of the off-target effects early on. You're not able to find new targets. If you don't have the transcriptomic, you don't see the full picture of those maps, right, that we talked about, the different layers. And DMPK, digital tolerability, matchmaker, all of those modules are important to get to the right molecule. So I just want to say that it's really, really important to have a collection of these algorithms. And they're not static. They're constantly evolving and getting better. And that's where the agility becomes very, very important. Because there's going to be new modules outside the 4 walls of Recursion. It's going to be very important to be open-minded and opportunistic to bring in what makes sense. All right. With that, I'm going to skip the phenome beta, but it's just great to see that some of these algorithms are actually being leveraged to shape the industry on the outside, not just keeping it inside. So what's next? Look, I think it's going to be really important to be careful and pragmatic as to where we go deeper. So a pipeline that's primarily based on molecules that are using generative AI and other approaches, but also the aspect around clinical development, right? Precision medicine, real-world data, accelerating our trial execution. So with that, I just want to close by saying it's been a great few weeks. getting to know the organization. I'm so excited to join in a couple of weeks. And I will say that, look, it's not going to be who has the best and the biggest data set. It's not going to be who has the best algorithm. It's going to be the person or the organization that can actually stitch it all together in a way to drive outcomes, which is about medicines. And I think that that's the ultimate focus that we all need to have. And it's exciting to see that here at Download Day and beyond with the teams. So thank you again.
Thank you, Nijhawan. And now to talk about partnerships, I'm going to bring up Matt Kinn. Matt.
Great. Thanks, Ryan. Thanks, everybody, for being here today. It's a pleasure to speak with you this afternoon. My name is Matthew Kinn. I'm Senior Vice President of Business Development and Corporate Initiatives here at Recursion. Great. So, as we've alluded to throughout the day, we have a three-prong approach to driving value here at Recursion. All good things come in threes. Of course, we have three of them. First is our internal pipeline, which is focused primarily on precision oncology and rare diseases. You heard about it this morning from Laura. You will hear more about it this afternoon as well. Second are broad research collaborations in complex therapeutic areas, such as our existing collaborations in neuroscience and undruggable oncology targets, as well as future collaborations that we might enter into in other intractable areas, such as cardiovascular metabolic disorders, inflammation, and immunology. Lastly, are partnerships and agreements related to data and models that underpin the Recursion OS. And you've heard about many of those models and data sets earlier today. Let's dive a little bit more into our partnerships. As a reminder, in December of 2021, we entered into a pioneering collaboration with Roche and Genentech. The goal of this collaboration is to identify and advance up to 40 therapeutic programs in the fields of neuroscience broadly, as well as a single oncology indication. Last year, we were very excited to announce that Genentech had exercised its option on our first validated hit series under the oncology arm of that collaboration, and we look forward to sharing additional potential future milestones related to both projects as well as map options here in the near term. So what's really exciting, I think, in addition to the progress that's been made to date, is actually the like-mindedness that we share with our Roche and Genentech counterparts. We're delighted today to be joined by John Marioni, who's going to speak a little bit more on this topic. In addition to having a distinguished academic career, John is the Senior Vice President and Head of Computational Sciences at Genentech. As well, John's a member of our Joint Governance Committee under the collaboration. So, John, I'll invite you up. Yeah, turn it over to you.
Thanks, Matt. So it's an absolute pleasure to be here. It's been a really fantastic day so far, learning about all the amazing work that goes on at Recursion, and seeing some of the examples and the demos is really cool. So I'm going to start with an old image. And this is just to say that this isn't new, what we're thinking about now. Technology has always driven our field. This is an image of a microscope from the 18th century. And Robert Hooke used it to look at slices of cork. And within that cork, he observed those structures. And that's the first use of the word cell, because it reminded him of the areas where monks used to sleep. So that's the etiology of the word cell, and the first time it was used as a microscope. But of course, we've been looking at cells as a field using various techniques for literally hundreds of years. And it's a reminder that as the technology evolves, the use of the microscope, the use of stainings, the use of single-cell genomics, it allows us to get more and more information about cells. So this is an image from some work of my own where we were looking at early development in the mouse context. And it's a reminder that technology, not just in the area of looking at cells, but in other areas, has really been a driver of biological discovery. It's been a driver of the work right across the field. And at the moment, we're on the cusp of another major technological leap forward, and that's artificial intelligence and machine learning and how we use that effectively. And that's going to be the next thing that's going to push this field forward, as we heard about so much this morning. But of course, it's not all solved, otherwise we wouldn't all be working on this, we wouldn't be thinking about this so much. We've got the ability now, in the labs that we can see to the left and all around here, to generate huge amounts of data from both healthy and perturbed conditions, but it's critically important that we make sense of these data, and in particular, can we predict perturbations that we've not seen? So in an ideal world, what we would like to be able to do is to predict in any cell type of our choosing what would be the impact of any perturbation, genetic or chemical, and we could then use that as the way to really motivate the experiments that we're going to do. This is not a trivial problem. There are tens of thousands of cell types, tens of thousands of genes. Once you start adding variants and think about different combinations of variants that act epistatically, this is an enormous search space. And compared to other areas of the sciences, so in physics or chemistry, the actual number of constraints is fairly limited here. We don't even know how to model fairly simple processes like transcription or the cell cycle. If we can't model that, then the ability to understand the impact of a particular perturbation in a complex multi-tissue organism is really tough. So we believe, and I think that this would be something that would be shared by everybody in this room here, that this is one setting where computational models, and especially foundation models and generative AI, can truly transform how we both discover and develop medicines. Now, as Nijhawan just said, there's a number of different facets where these approaches are going to be applied. I'm going to focus on the discovery angle here. But of course, it's also true that you could use these approaches to design better molecules. You start with information about structure, affinity, other binding properties, etc. And you could use that to search through different areas of the chemical space. And later on, when we think about designing trials, absolutely, but also in automating that process. Regulatory filings are slow. It's not glamorous. It's not going to improve your PTS, but it is going to decrease the time it takes to get from that molecule that works to a filing that's going to impact the lives of patients, which is what we're all here to do. So within GRED, so that's Genentech Research and Early Development, I lead the computational sciences organization. It's through this that I have such a great understanding and a wonderful partnership with colleagues here in Recursion. And within Genentech, we're the computational part of this that's making this vision a reality, from everything from infrastructure, data management, through to model development and application, all the way through to phase 2 trials. How? Well, it's actually a very simple and similar model to the one that we've heard a lot about today. It's something we call lab in a loop, you call it virtuous cycle or virtuous circle, but the idea, the concept is very similar. You start with an experiment that's able to generate very large amounts of data, and I do think that it's important to emphasize that the scale of data generation is really critical here. We've previously gone with very targeted data generation. That ability to sample randomly from across the whole space is really important because we don't know what we don't know. There's large areas of chemical space, but especially of biological space, where we just don't know what this perturbation is going to do. So that ability to really generate data, perturbation data from across that whole space is really vital. And having generated that data, you then use that to feed those models that are then going to be able to make the predictions, that are able to stitch the data together, and then to generate those new hypotheses that I talked about, that ability to predict what is the impact of a particular molecule in a particular cellular context. What's going to be the impact of that genetic perturbation, and in time, how do they interact with one another? Just a very simple example to illustrate why that ability to generate data matters so much. Here's some strings of DNA. Very simple problem. We want to take an 80-mer sequence of this, and we want to be able to predict what's its effect going to be. How expressed is that 80-mer going to be in a random context? Well, the way that you might typically have approached this problem in the past would be to design 80-mers, perhaps based around known genes, around known RNAs that you could use in order to test this. But actually, that's limited. And so what this is work from my colleague, Aviv Regev, that she did, she decided that instead of actually trying to synthesize in a targeted way particular sequences, they would just generate 10 to the 8 random sequences from that 4 to the 80th space. And they were able to show, using this, that they were able to generate training data, where they were able to get how expressed each of these sequence tags were, and then build an oracle-based model, just a relatively simple transformer, and using that, they were able to show that they could be able to predict the expression of an independent random 80-mer better than those models that were building upon known sequences, so the 10,000 or so synthesized sequences that people have been focusing on in the past. So really that combination of computational technology, 80-mer sequences that you could generate at random and at high throughput, and the ability to really work tightly between the different spaces, the wet lab, the dry lab, computational science collaborators on the underlying infrastructure, so important. But of course, that's a relatively simple problem. We want to scale this up to really discover new targets here. And we think about this across all aspects of R&D, where we go from the experiment to the data to the algorithm really quickly, and it's that tightness that matters so much. We've really got to have those readouts that are relatively simple to generate, and we've got to be able to generate them at massive scale, because we don't want to have a wonderful readout that takes 18 months to actually give you something that you could then feed back in. You've got to have the right readout that you can use at scale. That's going to enable you to generate those models that are then going to be useful. And of course, there's more to it than that. There's also these strategic partnerships. We share a partnership with NVIDIA, both Recursion and Genentech on the computing side. We, of course, have a great partnership together, and we have a variety of other partners in-house, including, I just want to emphasize, AWS here, with whom we work on quite a lot of the computing infrastructure also. Okay, so where do we hope this is going to get us? Well, we hope that it's going to get us, and I think this is a shared goal again, to that right target or chemical matter for the disease that you care about. So together with Recursion, we work with you guys in a couple of areas: gastrointestinal cancers, the first system, and then that very broad collaboration in the neuroscience space. And in each of these, starting with that cellular system, and I would just say that it is harder in the non-cancer space, it's something that's worth emphasizing, to have the right model system that you can use to actually generate the relevant data. That is just a challenge. But having generated that system, you can then target it with CRISPR, with known small molecule compounds, and you can see how it reacts. And you can measure that output in a number of different ways, for the cell painting over on the left, but also single cell transcriptomics. So perturb-seq-like assays, other assays that you could use to read out the impact of these perturbations. And having done that, you can build models. You can either build unimodal models that will consider one of the assays independently, so imaging and single cell RNA-seq, and then think about how to bring it together. Or you can build multimodal models, and those are really very powerful, where you're able to combine the different types of data together in a single model. We've heard already today about how that combination of data really matters. And then having done it, what we all want to be able to do ultimately is to be able to predict better targets, better compounds that are going to have the effect that we want in these cellular systems, bring that through into the clinic, test those, and fingers crossed, they read through positively and impact the patient's lives, which is what we really want. So just a little bit upon the model development, because there's a lot that's gone on in the sort of the data generation. We've heard a lot about that today, but of course, the actual modeling aspect here is very important as well. And in our collaboration, I mentioned that there are two facets to this. There's one where we kind of have independent modeling, where we'll model and generate the single cell transcriptomics data and combine that, but in a non-fully integrated way with the cell painting. And that's where you have this kind of independent phenomaps that you try and push together, but also the multimodal, where we're really working tightly, the computational scientists in both companies, to build and to bring these models to reality, where we're using a variety of multimodal embedding tools. There are a lot of these out there, I won't dwell on that, to build these phenotranscriptome maps. And we really already begin to see the power of some of these. So it's really exciting. We're really excited to build on this collaboration as we move forward. This is important. The models are only as good as the data. It's something that we just heard from Najat as well. There are challenges here. I just want to emphasize, from my perspective, what I see as four of the major ones, and I guess comfortingly, a lot of it overlaps. Data management, metadata, and access. There's a lot of data out there. I think historically one of the challenges that you will have in a large pharma is that maybe that infrastructure, that data layer, has not been as coherently set up as it could be. We see that, and that ability to really build that is something we're emphasizing heavily at the moment, to make that data accessible, and especially in a forward-looking way. Because, and a statistic that I always like is that in the next 2 years, we'll generate more data than the field has in the last 40. So we should be forward-looking here. We have to be thinking about how we grandfather over those valuable data sets, but we have to always look forward, because that's how we're going to push this. Integrating expertise from multiple disciplines. We've heard about the importance of these different disciplines. We also heard a little bit about the culture. That's very, very, very important, that these different groups feel comfortable talking to one another, that they feel comfortable working together, and that we do begin to understand and speak the same language. There has been a little bit of a history. I remember when I started my PhD 20 odd years ago, so quite some time in the distant past, there was definitely a sense that computational biologists were a service group. You would throw data to them, they'd generate something, it would be passed back over the fence, and someone with an understanding of the biological system would do something interesting with it. We've got to get over that. That's not where we need to be. We need to be fully engaged with one another right across these disciplines. If we don't do that, we're not going to take advantage of these tools properly. We're not going to be able to really grasp their full potential. I think that's still a challenge. If you talk to colleagues in other industries, they would say that 80% of this is a people problem. Really getting them to use the tools properly, getting them to use, we heard about Loo, right? That's the name of your system. You've got to not use it like Google. You've got to actually talk to it and engage in a conversation with it. And for many people, that's a very different way of thinking. We're learning a little bit with ChatGPT, other bots, how to engage like that. It's still not easy. It's still very much the tendency for many people to use this rather like a Google search engine. We're not going to get the most out of it. So we have to train, we have to learn, and we have to be respectful on all sides that all of these expertises, the basic science, the computational science, the infrastructure, and that collaboration externally is going to be necessary for this to succeed. Scientific computing, again, I think this is something that we would share, that need to access outstanding scientific compute is going to be absolutely essential. And maybe different models, maybe we need to have more bursty access to compute rather than access to just large volumes. And also there's access to the really very, very good software engineers that are able to optimize the code, that are able to make it run fast. These are difficult people to find. They're, of course, in huge demand across a number of industries. But if we are able to grasp that and to really exploit that and to work collaboratively, we do this in a part of our partnership with NVIDIA, that's how we're going to accelerate the models. And finally, democratizing access and ensuring use of the data and the models. And here I think that we look very excitedly towards autonomous agents, agent-based models as a way of being able to really democratize that access, where you have that garden of software, that garden of data sets, and the agents are able to reason about where to go, and they're able to engage with the users from all disciplines, not just those that have a computational background, to really dig into that data and to make the most of it. However, this is all worth it. It's hard, but it's going to be worth it, because I really believe that the impact of this, if we get it right, is going to be transformative. Again, transformative medicines is what we are, because transformative medicines change people's lives. At Genentech, we're very happy to work with all of you guys here. It's been a great partnership so far. I look forward to many more years of fruitfully collaborating. Thank you for the invitation. I look forward to the rest of the day. Thank you.
Spoken like a true recursionaut. Actually, John and I were joking earlier today when we were flipping through the slides, whether we should take out the slides that feel redundant. And it feels like the presentation would have gotten compressed quite significantly. I hope it's striking to everybody how similar the vision is across these two organizations. And I'll just say, like, on behalf of the Recursion team, like, it's a real privilege to work with you and your team. I think the vocabulary that we use, the way we think about problems is so, like, well-tuned with one another. And so again, we're looking forward to sharing more updates about the Roche Genentech collaboration in the near term. Thanks again, John. Turning a page, so in September of 2020, as many of you know, we entered into a multi-year collaboration with Bayer. And in November of 2023, just this past year, we transitioned that collaboration moving forward to focus on 7 projects of relevance to undruggable oncology targets. That work around this new reshaped oncology interest began in earnest in Q1 of this year. Here we stand today, here in June, the end of Q2, and I'm excited to announce that we've recently initiated the first joint project under this collaboration, which is now moving quickly towards lead series nomination. Additionally, we are well on track to deliver 25 multimodal data packages in Q3 of this year that will provide a lot more substrate for additional project nominations here in the near term as well. As you already heard about today, and I want to re-emphasize, we're really excited today, this morning with our 8K, and throughout the day today, you've heard that Bayer will also be the first beta user of our low LLM orchestrated workflow engine software. Low is going to get rolled out across the collaboration, and as I think you probably picked up on if you went to the demo, it offers a lot more of an intuitive and exploratory research environment for scientists on both sides of the partnership. So extremely excited to make that announcement today, and very excited to see where that goes with our collaborators at Bayer. Earlier this year, we had the privilege of hosting many of our Bayer colleagues here at our Salt Lake City headquarters. I think as many people in the audience can appreciate, it's a different feeling when you can actually walk around, see the robots, and the scale of data generation that we're able to produce here at Recursion. But that's just my words. I really want you to hear firsthand from our Bayer counterparts as to their reflections on the day.
Today was an incredibly exciting day for us. We got an opportunity to host our Bayer partners for the very first time.
This is a collaboration that has been ongoing for quite some time, but this aspect of the collaboration is new.
We're going to be able to explore a really large number of targets for oncology, which is something that traditionally we're not able to do.
We've made a lot of strides in cancer treatments over the past few decades. There have been several revolutions around immunotherapy, targeted therapy, angiogenesis. And looking beyond the lamppost, looking beyond the known biology is what Recursion's platform promises to unlock.
It's a new way of thinking about how to do target discovery in precision oncology.
The problem is really the on-target druggability for various reasons. So what I'm really looking forward is to explore novel biology associated with that target space.
It's really allowing us to do unbiased discovery.
Artificial intelligence relies on a huge volume of data, and that data is being generated impressively fast and at scale here at Recursion.
Obviously the automation is exceptionally cool, but it's the scale with which the automation is occurring. These are experiments that I think all of us understand how to do in very small ways, but seeing the scale with which you can do that and generate all of the data was really remarkable.
This partnership offers the opportunity, the best of both worlds, bringing the real technology forward and artificial intelligence leading edge emphasis of Recursion with the legacy of drug development from Bayer.
It's always easier when you let the partners do the talking for you. So in addition to our industry-leading collaborations with pharmaceutical companies, we have and we will continue to partner with leading companies in the technology and the data ecosystem as well. Reference has been made throughout the day to our invaluable collaborations with Tempus and with NVIDIA. Additionally, we have formed a very deep relationship with our partners over at Innovating, a Ukraine-based chemical synthesis CRO. We've brought our software tools, many of which you've heard about today, to the Innovating collaboration, and collectively we've been able to more efficiently explore this massive chemical universe that you've been hearing about. And also we're working towards developing novel compound libraries, both for Recursion's benefit, but also for Innovating customers' benefit as well. Additionally, I want to highlight one agreement that we entered into with Helix just a few weeks ago in May of 2024. Helix is a leading population genomics company that's enabling health system partners, life science organizations, and payers to accelerate the integration of genomic data into both patient care and therapeutic development. Well, I'm really excited about it. Helix is building this sustainable model of generating clinical genomics data by pairing Helix's whole exome sequencing platform with standardized EHR data across a really diverse network of health system partners. They're shown here on this slide. Representative examples of those health system partners include St. Luke's Health, WellSpan, and many more. Collectively, these entities have committed to contributing 1 million clinical genomic records over the next 3 years, which make this an extremely rich source of data moving forward. At Recursion, and under the terms of our agreement, we have access to hundreds of thousands of these unique de-identified clinical genomic records every single year. And we look forward to using that data in order to train more causal AI models like you heard about this morning, to design biomarker strategies, patient stratification strategies, and ultimately identify novel targets and medicines to impact human health. So in wrapping up, this three-pronged approach is really driving value continuously at Recursion, with leading collaborations across pharma and the technology ecosystem. We believe that we are incredibly well prepared to accelerate our mission to impact human health at scale, and I very much look forward to sharing more updates about all of our collaborations in the near term. Thanks so much.
Thanks so much, Matt. And now I am excited to introduce Dr. David Mauro, Chief Medical Officer of Recursion. Dave.
Thank you. Good afternoon, everyone. I've often been accused by my colleagues of my dispassionate communication or about the pipeline, dispassionate but objective. So I'll try to amp up the passion, but I will be objective. What I'm going to talk to you over the next half hour or so is I'll walk you through each of the programs. I'll talk a bit about the indication. I'll spend a little bit of time talking about the insight and then how we validated the insight. And then I'll spend the majority of time for each program talking about the trials with really an emphasis on what are the expectations, what does good look like. So with that, let's get into the pipeline. So this is the pipeline. This is a robust pipeline. As you can see, we have 4 ongoing phase 2 studies. We have a phase 2 study that's starting in Q4 of this year for our C. diff program. And you heard a little bit about the INDs. The RBM39 program Laura mentioned earlier. We expect to submit that IND in Q3 and initiate the study in Q4. The Epsilon program we plan to submit early in Q1 of 2025. So what you heard about this morning was how the company is rapidly translating insight into clinical programs. And this is the evidence of that here. This is a monumental amount of work. We expect that this pipeline will increase. But what it seeks to achieve over the next 18 months are 7 readouts of both phase 2 and phase 1 data. So with that, let's get into the first program. So this is our REC-994 for the treatment of symptomatic cerebral cavernous malformation. So this is probably the largest disease that no one has heard about. It's certainly a disease that I was unaware of before I came to Recursion. So believe it or not, there are over 1 million patients that suffer from this disease. About 1/3 of them are symptomatic. Symptoms can range from mild headaches to severe hemorrhagic stroke, right? So there are no approved therapies, and there's been only one completed phase 2 trial run by a relatively small investigator-initiated group. So most patients are either observed, their symptoms are managed through over-the-counter meds, or they're offered surgery, in some cases radiation therapy. But that's really dependent upon a lot of things, right? How bad are the symptoms? Where are the lesions? If these are deep lesions, it's going to be really, really hard to go after. So the unmet need is significant. At the heart of the pathology is altered endothelium, that leads to vascular malformations. Our therapy targets the endothelium. So a question that I've heard, and Michael has heard, and others have heard, is if this is such a problem and there are so many patients, why aren't more companies going after this disease? Well, look, I can't speak for other companies, but I can kind of speak to from a biologist perspective, and I sort of pretend to be a biologist at some time in my life. You know, the biology here is very complicated. You have multiple proteins interacting, converging on multiple pathways. So the target is unclear, right? These are the types of biology and indications that lends itself to the Recursion OS, right? So we can go after, in an agnostic approach, go after looking at many different molecules and try to come up with the one that fits best. So this is a program that has sort of founded in the roots of the company, and we're very excited about the readout coming in Q3 of this year. So I've talked a little bit about the insight, right, where this came out of. This is some preclinical data that's been around for a while. This came out of our founder's PhD dissertation. And it covers a couple of panels. The top panel looks at 2 CCM knockout models. And what you can see as compared to control, we're able to reduce the lesion burden. So some of these patients have multiple lesions, right? 5, 6, 7, and others have few. But in both cases, both the size and the lesion burden were reduced. We think perhaps what's more important and might be clinically relevant is shown on panels 2 and 3. We think inherently this is a disease of the vascular permeability. And we think that our therapy, at least in these models, showed that we're able to impact, you know, the permeability. So this is something that we can monitor, measure in the clinic. So this is the study design. So we've enrolled, and this study happened to enroll relatively fast. I think we were able to enroll 60 patients in somewhere around 9 months or so. So really speaks to sort of the unmet need and the desire for patients to get treatment. So all patients will have MRI-confirmed lesions. There is a familial component of this disease, which accounts for around 20 to 30% of the patients, and then there is a sporadic form. We're enrolling both patients. All patients need to have symptoms that the clinician felt were directly related to CCM. So these are not patients that are walking around that are asymptomatic. I'll talk a little bit about the primary and secondary endpoints in a minute. You can see the schema. It's a randomized, blinded study. Patients are treated for a year, and then they're given the opportunity to go on the extension study. Most of the patients, I think to date, there may be only 3 patients that have not moved on to the extension study. So by and large, patients are continuing on study, and we will follow those patients as they progress in their therapy. So why a year, right? Why not 6 months? Why not 3 months? Well, this is a slow-growing disease. Patients often live with this disease their entire life. So input from KOLs, it was felt that 12 months is sort of the minimum amount of time that you would want to treat and treat these patients. So obviously, we're going to be treating these patients. We will continue to treat them in the extension phase, and we will be monitoring their therapy. What are we looking for? This is the question that we get asked again and again. So this is not a response rate study. This is not a PFS study, right? The primary objective of this study is safety and tolerability. And when I've talked about, you know, the primary being safety, I get a lot of eyes rolling. Yeah, who cares? It's important, right? It's important to not only have a therapy that's effective, but it's important to have a therapy that patients can take for a long time. An effective therapy that patients can only handle for 6 months is not going to be one that's used, right? So we want to be able to evaluate the safety of these patients for a long period of time. Now let's talk about the secondary and exploratory endpoints. So why wouldn't you elevate efficacy to a primary endpoint? The answer is we simply don't know enough about the disease. We don't know enough about it. KOLs don't know enough about it. The FDA doesn't know about it. There's been no real data to say what efficacy endpoint is important. So we're not going to sort of fictitiously power a study around an endpoint that we have no data. So this, in essence, is an exploratory study. But what are we looking for, right? What does good look like? Well, it's shown here, really. Our analysis is going to be looking for multiple trends across multiple endpoints. I've talked about permeability, looking at hemosiderin deposition. I've talked about changes in lesion burden. Subgroup analysis is going to be really, really important because this is an inherently heterogeneous disease. You have some patients that have come on study that have had hemorrhage. You had other patients that have come on study that have big lesions, others small lesions, others with headache. So I think it would not be prudent for us to just sort of go after an efficacy endpoint. But what we're trying to find are trends that are meaningful for us, trends that are meaningful for KOLs, and most important, for the patients. Our hope is that if we see these compelling trends, we're going to be able to make a case with the FDA on what does your next study look like, and whether that be a 2-3 or right into a 2-3, right into a 3. So I think we're very excited. The team has done a lot of work. This has been a well-run study. Investigators and patients have been very passionate. And I just want to acknowledge Connie Lee, who leads the patient advocacy group for CCM. Any questions about CCM? Ask her. She knows more about it than me. So our next program is our 2282. This is our HDAC inhibitor for the treatment of progressive NF2 mutated meningiomas. So a little bit about the disease. So NF2 meningiomas sit within a constellation or a syndrome known as NF2-related schwannomatosis, right? So within that disease, you have meningiomas, schwannomas, ependymomas, meningiomas. It's a complicated disease, and not all the tumors are alike. You can just imagine what this disease is. Patients suffer from multiple tumors that are benign. The standard of care for the treatment of this patient is surgery or radiation, but this is a relentless disease. You cut out the tumor, it comes back. You radiate the tumor, it comes back. You shrink the tumor, it comes back. So clearly, a therapeutic option for patients who don't have to go through 7 surgeries like this patient has is critically important. I'll talk a little bit about sort of the endpoints. What the FDA has communicated and what the field has tried to show was that you really want to delay progression. So we'll talk about sort of the endpoints that we'll be evaluating in our study. So you've seen the panel on the right. I'll just briefly frame it up as insight on the right or on the left, validation on the right. So we were not looking to build an NF2 program. This is insight, novel insight that came out of our platform agnostically. And you can see the validation in what is considered a clinically relevant FAP model. Rather striking, prevents growth, and also what's probably most important is the regrowth, right? Again, these tumors come back, so simply just decreasing their size is only half of the equation. You not only have to manage the tumor, but you have to prevent regrowth. So, this is fairly compelling data, along with all the other work that we've done. We've translated this into our study. So there have been a lot of molecules that have been tested in NF2 disease, right? I think one of the challenges that these clinical trials have faced is the heterogeneity of the disease. So this is a disease that waxes and wanes. Patients have progression and then they become indolent. Patients have progression and then they become indolent. And you're also mixing different types of tumors together in a trial. So what we've tried to do in this study is control for that, right? So we're only evaluating meningiomas. Obviously, if we see a signal, it'll give us reason to believe that we might be able to go after other tumors. And the other thing that I think is probably more important is we've tried to control for progression. What you don't want to do is enroll patients that have relatively indolent disease, and then it's difficult to interpret their progression-free survival. Is it just because the patient simply doesn't have aggressive disease? So what we tried to do, and the advice that we've got from our KOLs, is try to enroll patients that have more aggressive disease. So these patients will have had to progress over the last 2 years, so we can kind of understand their slope of progression prior to coming on study. And obviously, what we're trying to do is decrease that slope of progression. So these are one of the endpoints that we'll be looking at. We'll also be looking at, you can see a futility analysis of PFS6. We've enrolled patients that have sporadic disease and familial disease. We'll be evaluating them together as well as in separate cohorts. So essentially, we've tried to control for things that other trials have not controlled for. You can see that they're dosing across 2 different dose levels. We'll get to this PFS analysis. We expect to have top-line data by the end of this year. If the data is encouraging, then the FDA said, come back and we'll talk about the next part of the study, which would likely be a controlled study that is randomized. So stay tuned for this data. Our next program is our MEK inhibitor. So this MEK inhibitor is being evaluated in 2 studies. One is an FAP study, which I'll talk about here. Another is our study in Axin 1 APC, which Nijhawan mentioned. So I hate to sort of characterize diseases as bad or worse, or this is a tough disease. Most of the patients that are diagnosed with this disease are diagnosed at a relatively early age. Often in their teens, it's an autosomal dominant. This is characterized by, if you can imagine, hundreds upon hundreds of polyps in the colon, right? Just imagine that. Patients are typically diagnosed in their late teens, early 20s. The treatment of choice is colectomy. So imagine having that as a 20-year-old, right? The risk in patients that are not resected, the risk of cancer is 100%, right? So even though you remove the colon, you still have polyps throughout the small intestine. These patients need to be monitored throughout life. And they're often managed through surgery because there are no approved therapies. So it's unfortunate, but over time, you're having more surgeries and patients are becoming more restricted. There's more morbidity. It's a horrible, horrible disease. So really what we're trying to do in this study is take some of the insights that came out of our OS, similar theme, and validate them. So this is another clinically relevant model, and I'm just showing you one slide. It's late in the day. I don't want to bore you with 1,000 slides, but this is fairly impressive data. In comparison to controls and other drugs that have shown, celecoxib has shown a decrease in polyps, we have a nice dose-dependent impact on both polyp count and high-grade dysplasia. So again, compelling data that we simply can't ignore. The study is as follows. Because this molecule has not been evaluated outside of cancer patients, we felt that it was particularly important to take a conservative approach fairly early on in the study. We really don't understand what the removal of a colon might have on the PK. So what we're doing here is a short dose escalation study. And once we have the recommended phase 2 dose, we'll expand it into a dose expansion. So what does good look like here? I think for us, what good looks like is a reduction in polyp burden. Can we shrink the adenomas? Can we overall reduce the number of adenomas? We think this is an important signal-seeking study. If we can show activity, then I think there's reason to believe that we could move this forward. But I think this is a sort of a succinct way to design a study and really understand whether or not if you have a drug that you can take further. So this study has been initiated. We are enrolling patients. We expect to have preliminary data by the first half of 2025. Our next program is with the treatment of solid tumors with AXIN1 or APC mutations. So if you look on the right, people have known about AXIN1 APC for years. This is not anything new. AXIN1 APC sit in a cluster of proteins that regulate the Wnt pathway. And people have known that there have been mutations within these genes for years as well. But it was always felt to be an undruggable target. Until you can really drug the target, you really don't know the clinical relevance of AXIN1 APC mutation. So right now, a patient comes in, they're screened with NGS, they have an AXIN1 APC mutations. There's nothing that the physician can do about. They'll go on standard of care. But we think our insight here is really novel, and there have been MEK inhibitors around for a long time. People know a lot about them, but this is really, unique insight that came out of Recursion. So here you see sort of the phenotypic screen, and what you found is that this particular MEK inhibitor, and it's this particular MEK inhibitor, not a class effect amongst all MEK inhibitors, was really phenotypically opposite to AXIN1 and APC mutation. So, okay, interesting, right? What do we do with that, right? Well, let's try to validate it in some models. And here you see on the right, we've looked at the relationship of targeting AXIN1 and APC mutations versus wild type. And we see a statistically significant improvement in both tumor growth inhibition as well as progression-free survival. So again, another insight that we think that we know is unique to Recursion. The trial is a fairly standard Simon two-stage design. We're enrolling patients with AXIN1 APC. We'll do a quick safety run-in at 12 mg, and we're looking for response rates. If we see one or more out of the first 10, we will continue to enroll. Nijhawan alluded to some of the approaches that we've taken to enroll patients. I think we have a very aggressive strategy, and this study is enrolling briskly. So this study is ongoing. We expect to have the phase 2 readout in the first half of 2025. So you start to get a theme, right? Lots of readouts. We've talked about it a lot, but this is a pipeline that is producing, hopefully, meaningful and positive clinical data. Our next program is with our toxin inhibitor, C. diff. So I don't need to explain to anyone in the room that C. diff is a problem. It's a problem here. It's a problem worldwide, right? Patients die from C. diff, right? This is not something where you just simply throw antibiotics at it and it goes away. And in fact, probably about 30% of the patients, 20 to 30% of the patients recur, and not just recur once, right? Recur twice, 3, 4, 5 times, right? So optimal treatment to C. diff should really factor on 2 approaches. One is to kill the bug with antibiotics, and that is fairly effective. The other is using an antitoxin, right? Because it's the toxins that produce the pathology, it's the toxins that produce the symptoms, and it's the toxins that predispose patients for further recurrence. This is our first NCE that we've taken to the clinic. RBM39 hopefully will be our second. Epsilon will be our third. Starting to see a theme, right? So the study, so this is some preclinical data. Again, insight on the left. But I think this data is fairly compelling on the right. So this is a well-known C. diff hamster model. Hamsters are infected with C. diff. They're treated with, in this case, 4 options: vehicle control, vancomycin, or in this study, our antitoxin versus the only other antitoxin that's approved, bezilotoximide, which is an IV infusion. So as you would expect, vanco does really well, surprise, surprise. But I think what everyone can see is a rather striking and significantly difference in probability of survival between our molecule and other antitoxins. So this is a program that is gathering a lot of interest among the community. Patients that have at high risk or have recurrent disease are often given antibiotics, and they're offered either a fecal transplant, which is not often readily available, or bezilotoximab. So we think there is an opportunity to impact here. The trial is straightforward, right? So we've taken patients that have high risk of recurrence. There's a fairly standard algorithm to identify those patients. They'll be treated with standard of care vanco. My sense is that most, if not all, of the patients will have symptom resolution. Then they'll be randomized to high, mid-dose, or observation. We expect that patients in the observation arm, based on real-world data, should start to recur within 4 to 6 weeks. So what we're trying to do is delay and improve on the rate of recurrence. So this study is expected to start in Q4 of this year, and we're very excited and hopefully have preliminary readout by the end of 2025. I'll quickly just walk you through, you've heard a bit about our RBM39, another NCE. This is the study design that's hopefully starting up by the end of the year. Fairly straightforward, phase 1, and then we'll get into dose confirmation. So this will be a study in biomarker-selected, relapsed, refractory, HR proficient solid tumors. We have not given any guidance on what the biomarker selection criteria is. As we get closer to the study, we will be providing that information. But again, we'll be using a lot of the same tactics to enroll patients, and we expect that there'll be a lot of enthusiasm. Hopefully, this study will enroll quickly. So let me switch gears a little bit. I'm not going to walk you through this slide. You guys have seen it. It's even too complicated for me. But look, at its heart, right? The essence of what this is all about is moving really rapidly from insight to IND. So when I interviewed here, Chris Gibson sort of challenged me. He says, well, what are you going to do to be innovative? We're innovative. Look at all this great stuff we're doing. Clinical development, it's a wasteland. There is no innovation, right? It's where efficiency goes to die. That's clinical development. So I said, Chris, stop bothering me. But more importantly, clinical development is less about innovation, in my opinion, and more about efficiency, right? There are a lot of tools out there, right? We cannot take 6 months to go from IND to first patient, first visit, right? And it's less about speed, right? We are regulated to go at a certain speed, but it is about efficiency, right? So what are we doing, right? So Chris is often challenging me, what are we doing to be more efficient? So, I mean, here you see just a few of the steps, right? So, look, I have to be efficient because as the recursion engine starts to pump out molecules, if I haven't taken a more innovative, efficient approach, there's going to be a backlog. And the clinical development program will sort of die in a quagmire, right? So some of the things that we're trying to do to make things more efficient, right? Look, one of the big problems that we have in the IND process is a very long GLP tox. It takes a month to run this study. It takes 4 months to get the data, right? So that's 3 months where we're sitting around not doing anything. So Recursion is constantly trying to think about ways that we can, I don't want to say get around it, but use our models, use our in silico testing that maybe start to move away from traditional GLP tox studies. Obviously, this is a conversation that we need to have with the FDA, but it's an important conversation. It's not one, we can't do standard practice. It's not going to work here, right? Manufacturing is rarely talked about, but it's one of the biggest black holes of money and time, right? The traditional approach is really built around making very large-scale amounts of drug, holding it, right? That takes a lot of time and effort. What we're thinking about and what we're doing is making quicker, smaller batch that can meet the needs of a trial within months of when we start the trial. We don't have to sit around. I've been in many trials in my life where I've sat around for 6 months waiting for drug supply. That's not going to happen here at Recursion. Biomarker patient selection strategy, you've heard about it, right? We cannot run trials where we're enrolling all comers, fingers crossed, hope we get a response, and let's see if we can deconvolute in the clinic. You can't deconvolute in the clinic. You have to come in with a bet, make the bet on the hypothesis, and if you bet right, you've won, and the patient has won. Nijhawan talked about real-world data. We're moving away from control arms, or we're really moving towards an effort to utilize real-world data to understand our controls. And the FDA has bought in on that. So what does that translate into? So instead of running a 3-arm study, now you can run a 2-arm study. What patient wants to be randomized to the control arm? I don't. Doc, give me the investigational drug, right? So now we can use real-world data to build, synthetic control arms, which will translate into getting data faster. Nijhawan talked about trial startup. The current model and where the industry is trending is really having networks using just-in-time. And then lastly, enrollment, right? We need to identify patients months before the trial is even started. So when the trial is started, both the site and the patients have options. So what I tried to communicate to you, at least on these last few slides, is the clinical development team is desperately trying to keep up with the rest of the company. And I think where we will see tremendous strides is being able to have study startup that is in a few months rather than half a year. So with that, I will stop. I appreciate your time.
We are going to bring up Michael Sikora, our Chief Financial Officer, to talk a little bit about finances and running the company. So, Michael, please. Thank you for your flexibility.
Thank you, Ryan. Thank you for that introduction. Thank you for everyone for being here at our download day, for unpacking the totality of this value proposition, and sitting in the seats that we sit, seeing Recursion the way we see it. You're all Recursionauts for a day. I'm Mike Sikora, CFO of Recursion, and I'm happy to be talking to you about our company, how we run this company, and the milestones that we have coming in the near term. A lot today you have heard about the what Recursion does and the why we do it. The what being the objectives, the goals that we have achieved and those that we are looking to achieve, and the why being the impact that we want to have on patients' lives. And I want to chat with you now about the how. How does Recursion operate? At Recursion, we believe that our culture and our people are key to driving value. And key to this message, key to this thinking is the Recursion mission: decoding biology to radically improve lives. And when I think about those 6 words, what I see in those 6 words is the ethos of a tech bio company. It is about the invocation of technology to understand a complex system, biology, to do what? With that understanding, find insight. With that insight, translate programs. With those programs, bring potential medicines to patients and have impact on society. I see what we do as one of the great scientific and societal challenges of our time. Now, in service to that mission are the Recursion principles and the Recursion values. These principles are the mindsets that we adopt for ourselves and our teams and how we try to organize around our work. These principles are explore the uncharted, create virtuous cycles. You've seen that many times today. You've heard John talk about that today. Build connected data, industrialize to scale, optimize for the portfolio, and challenge assumptions. If we think about the values, these are the behaviors that we want to adopt for ourselves, that we want to model for ourselves, for our teammates. And as you look around, you see these words written on the very walls. Together, these principles and these values, you have seen in all of the content, the state of the company, the OS, the preclinical, the partnerships, the state of the industry, the clinical. These principles and values are reflected again and again. And it is in these principles and in these values that we want to drive the mission forward and have tremendous impact in patients' lives. Chris, Nijhawan, others have talked about the fundamental ingredients for a tech bio company. Of course there is data. Of course there is compute. But people. People are the essential ingredient here. At Recursion, we have over 500 employees. Approximately 40% are technologists, data scientists, machine learning scientists, automation engineers. Approximately 35% are life scientists, biologists, chemists, folks in clinical development. And approximately 25% are strategic operations. When I look at the demographic of Recursion, approximately equal parts of life science and technology, I see these two worlds of technology and life science coming together, creating a unique culture. And that is something that we strive to have here at this headquarters and in the other locations that we have, such as in the San Francisco Bay Area, our office in Toronto, our office in Montreal, and our new office in London, where we are finding great talent, all in service to the Recursion mission. Throughout the day, you have heard us talk about the Recursion operating system, the Recursion OS. And that OS is the process for how we conduct drug discovery and development, where we move an insight into validation, into optimization, translation, and onto the clinic. When we think about our operating model, how we organize ourselves, we want to organize ourselves in line with our work, that very drug discovery process. And in so doing, we become part of the process. We industrialize ourselves as we industrialize that drug discovery process. At Recursion, there are 4 primary groups of how we organize our work. There is the Inception valence groups, which de-risks novel capabilities to expand the Recursion OS to a point of a minimum viable product. You can think about this group as operating in a way that we're creating new 0 to 1 shifts in technology. There is the OS group, where we are developing, deploying, and improving the platform and workflows that make up the Recursion OS that you heard about today. You can think about this group as then taking that one solution to a many solution and scaling it repeatably so. From there, there is the value group, which translates data and insights into value for our patients, our partners, investors, and other stakeholders. You heard from Matt about the business model that we have, how we are translating insights across the 3 primary axes for how we drive value across our pipeline, our partnerships, and our data. And lastly is the accelerator group, where we are driving company-wide efficiencies and paving the way for achieving the Recursion mission. Here, this group is finding efficiencies within each hub, between each hub, and across all of the hubs. Throughout today, you have heard a good bit about the milestones that we have coming in the near term. I want to now summarize all of these for you across the pipeline, the partnerships, and the platform. Earlier today, you heard from Laura, from Dave, as they unpack the pipeline in the context of the preclinical and the clinical programs that are coming. We have 7 clinical trial readouts expected in 18 months. I don't care if you want to style yourself as tech bio, biotech, or some other name. I see that as an extraordinary amount of data that's coming. I'm very excited about that. With respect to the pipeline, for cerebral cavernous malformation, we have a phase 2 readout expected in September of this year. For neurofibromatosis type 2, we have a phase 2 safety preliminary efficacy study expected Q4 of this year. For familial adenomatous polyposis, we have a phase 2 safety preliminary efficacy study expected first half of next year. For our Axon 1 APC mutant cancer program, we recently, in Q1, had our phase 2 FPI, and we expect to have safety preliminary efficacy data also first half of next year. Lastly, on the phase 2 side, we have our program in C. difficile infection, where we expect a phase 2 initiation Q4 of this year with a preliminary readout expected by year-end next year. That's 5 phase 2 readouts that we have coming. Behind those 5 phase 2 readouts, we have our program for HR proficient cancers. This is our target RBM39 program, where we expect an IND submission Q3, so next quarter this year, with a phase 1/2 initiation expected in Q4 of this year, with phase 1 dose escalation data by end of next year. Also, there is our Target Epsilon program. This is our novel target in fibrotic disease, where we expect an IND to be submitted early next year with phase 1 healthy volunteer data by end of next year. Those are the 7 clinical readouts that we have coming in a year and a half. And behind all of this are dozens of internal and partner programs in early stages, with our first LLM and causal model-driven programs now entering the clinic that you heard from Laura. Now let's talk about some of the partnerships and some of the platform milestones. You heard Matt talk about the partnerships and how we're advancing that work. On the Roche Genentech side, last October, we had a validation program option exercised for our first validated hit series in oncology, and there's the potential for program and map options in the near or very near terms. With respect to Bayer, we have already delivered multiple oncology data packages, and we are on track to complete 25 data packages in Q3 of this year, so next quarter. We've already initiated and are advancing our first joint program towards the lead series nomination, and there is the potential for near-term program options. As we've heard here today from a number of speakers, we are happy that Bayer has agreed to be our first beta user of LOW for this partnership to do drug discovery and development together. With respect to our partnerships in Tempus and Helix, here we are building large-scale causal AI models to generate target hypotheses across cancer as well as other disease areas, and we are already starting to explore novel targets related to non-small cell lung cancer. Behind all of this, there is the potential for other additional partnerships in large, intractable areas of biology. On our platform side, you've heard about all the great work that we're doing here from Lina. We're very excited to now have built our first genome-scale transcriptomics map, and now we start to move towards a multi-omic foundation model as you saw that coupling between phenomics and transcriptomics. We also have been developing active learning, and you've seen how we have now been deploying that active learning to build maps, to enrich maps, not just in some of the cell types we already have, but in new cell types, in new kinds of omics. And we're looking forward to exploring this technique in other kinds of omics data, like proteomics, organoids, spheroids, and with automated synthesis. There's also the potential to make some of our data and tools available to pharma customers as other commercial users. And you've already heard how today Bayer is going to be our first beta user of Low going forward. As you think about all of the work, all the great work that's been putting into the Recursion operating system to evolve it, what I see when I look at the Recursion OS, the three things that I see Recursion doing well. Recursion is very good at integrating a new technology, scaling that technology, automating that technology, doing it again, doing it again, doing it again. And as we continue to follow this approach, I see us continuously wading into autonomous discovery, and I see us operating this way from a strong financial position as we drive value across our pipeline, our partnerships, and our platform.
Thank you so much, Michael. Ladies and gentlemen, and our esteemed guests, I am so excited to bring Chris Gibson, our CEO and founder, back to the stage, and then Jensen Huang, the founder and CEO of NVIDIA. Welcome. Take a seat.
Well, thank you so much. I know you flew out to be with us today. It means the world. We've been getting to know your team for years. Kimberly, Rory, Anthony, the whole team. They're just incredible. Yeah. And it means the world to us that you're out here to talk about biology with us today.
This is my team. I think this is the largest investment we've ever made outside of our company.
Well, thank you. That means the world. When we were speaking last, you talked about, at the beginning of your career, how the silicon chip industry was laboratory-based, empirical-based, and you saw it transition to entirely in silico, or almost entirely in silico. What lessons can we learn in the biology field from that transition? Are there parallels? Can you talk a little bit about that?
There are absolutely parallels. And this is my career started 40 years ago, 41 years ago. And it was right at the time when computer-aided design started to make progress. Not that people didn't talk about computer-aided design before. It was just the combination of algorithms, computers that are fast enough, and know-how. Now that know-how was described as, and introduced this word into modern chip design that was really, came out at that time. And that word is called methodology. Until then, nobody used the word methodology. And methodology was inspired by Mead and Conway. I don't know if you guys have ever seen that, read that book, but it was written by a professor at Caltech. And anyhow, two of them, Mead and Conway, wrote this book to describe how you could create using very simple methodologies. It was based on first principles, but ultimately simplified the chip design methodology that enabled the creation of very large chips. And it's a book about VLSI design, VLSI systems. The concept that finally chips were able to be sufficiently large and complex that you could capture entire systems within it. And so they described methodologies for designing, for laying out the transistors, and how to simulate it, how to scale it. Mead and Conway's book really inspired a whole generation of chip designers. So the three things came together: algorithms, and now this is happening in your industry. Computing, this is happening in your industry, and know-how. And these three things, of course, in the case of chip design, the know-how doesn't as much involve the amount of data that your industry requires. But if you look at the foundation of Recursion, these three things are at play. The invention of a new algorithm, or new family of algorithms that we call deep learning, supercomputing capabilities that you've used here, and that we partnered with you to create. And of course, this know-how from robotics laboratory to generating and systematically generating data to learning from it, all of the multimodality nature of biology, and the know-how of processing, extracting the meaning of biology that's embedded within life, within that data. And so this know-how, this combination of all these things are happening right here. But I had the benefit of seeing it for the very first time in chip design 40 years ago. And the amazing thing is my manager spent all of his time in the lab. I was the first generation that was comfortable in the lab and not in the lab. We don't have a chip designer today that spends any time in the lab. They never go to the lab. The only reason to go to the lab is really just for the party. You know, the chip came back and is working exactly as we expected. Now, you have to imagine this. 10,000 engineers work together for 3 or 4 years at a time. They take all of their work, they put it into one chip. That chip comes back in a massive system. Thousands of these chips together, a whole lot of different types of chips, and we turn it on. It's an insane idea. And on the day that the computer comes alive and is working, it's not a miracle to me. It's fully expected to me. In fact, it's just another day in the life of that chip. And the reason for that is because that chip has existed for a long time in silico. Mm-hmm. For a long time, it's been doing the job that it was intended to do and it was just doing it inside this chip is doing it all inside another chip that we had built last time. And so you got these inception levels, chips that are creating next generation chips so that we can design algorithms to design next generation chips.
Recursive in that way.
Yeah, exactly. It's recursive in that way. It's almost like recursion. But that's it. That was my generation. I got to see it.
Did it feel inevitable at the time to you, to everybody else, that progression, or did it not?
Most people at the time would tell you that it will never work, and the reason for that is because the corner conditions, all the long tail of problems, and all the pain and suffering that they endured in the laboratories, and all of the chips that came back completely non-functional, their life experience caused them to not believe it's possible. And I think the journey of almost every industry is kind of the same. That the early people who made that industry suffered so greatly and things worked so poorly that by the time that it was ready to work well, they don't believe it. It can't possibly be that easy. Now, there's nothing easy about it, of course. It's just that the learnings were codified into tools. And in our case, and this is the part that's hard for you, and the reason why it's taken so long, in our case, we can change our, if you will, our transistors are, the minimum unit of our biology, okay? And we could change the structure of the transistor until we could design with it. You can't. You know, your transistors you have to live with. They are what they are. Our transistors, we shape them until they behave the way that we expect, or that they fit within the distribution of the capabilities of our simulation capabilities. And so if we can't predict how a transistor or a chip behaves in these corner conditions, well, don't go there. That was the easy thing. We shift our design rules. That's why we have these things called design rules. You don't have design rules.
Unfortunately.
Life is the rules, you know, and evolution is the rule that you live with. We have the opportunity to shape our transistors and our chips to the point where it turns out our transistors are so small now, they're statistically different. And if one transistor is pointing this way, another transistor is pointing this way, they behave slightly differently. Well, the answer is very simple. Make them all point the same way. And so we cause our chip designs to do things so that they behave according to things that we understand until we could push the limits further. And so otherwise we have these things called design rules and methodologies, and then everything kind of fits within it. Your challenge is much harder because you have to learn the behavior, you have to learn the meaning, the behavior, the properties of these biologics just the way they are. And well, the good news is finally you have the technology necessary to do that.
That's right.
I think we, within our grasp, it surely feels that between the robotics laboratories that you're creating for data process, data collection, systematic data collection, and machine learning, and the supercomputers that we build together, you might be within a click or two away from really being able to understand the meaning of life.
That's a tall order, but we're working on it.
Yeah.
When you look at problems to approach at NVIDIA,
You're kind of like where the first yellow.
Where the first yellow
is,
holds?
yeah.
Okay.
You're close.
For those who can't see, that's almost to the top of the wall. For clarity, for the investors.
That's where
in the
you guys
audience.
are. First.
You are one of the few companies that gets to work across every industry, basically.
Yeah.
When you look at your stance in healthcare, I've heard there's sort of 3 rules. You ask whether a problem is hard, you ask whether NVIDIA can make a unique contribution, and you ask whether that will be impactful. I think it's easy to imagine that life sciences, healthcare is hard. It's easy to imagine it's impactful. What is the unique contribution that NVIDIA can make in the context of healthcare? What is your stance on healthcare broadly, beyond Recursion, just zooming out?
Yeah. Well, the 3 things that I said, just to flip it on its side, the alternative is go do something better that somebody else has already done. Pursue a path that leads to the fastest ROI. And so these are the characteristics of people who love to win and make a lot of money fast. And there's nothing wrong with that. There's nothing wrong with any of that. But that's not us. Our personality is to go do something nobody's ever done before. Do something that if we didn't do it, nobody's going to do it. And 3, if you choose well, not only will you enjoy the journey, you can make a real contribution. And you might be able to live a life of purpose. And so that's kind of in our DNA. That describes what NVIDIA is. It describes the way I talk to the company, the challenges we select, the way we approach opportunities and threats and challenges and things like that. And so I think that's in our DNA. Now, where the problem, of course, of understanding the language of life and to be able to do drug discovery in silico, if that's not a hard challenge, I'm not sure what is. I mean, that's an insanely hard problem. However, it's within our lifetime to do something about it. And as you know, earlier we were saying that there are 3 ingredients. There's the ingredient of the algorithm, there's the ingredient of computing, and there's methodology, which in a large scale is kind of domain expertise. Notice we could contribute to 2 of those 3 things. in a pretty profound way, you know, in a really deep way. And because we don't have that domain expertise and we don't desire to have that domain expertise, we could be a great partner to somebody. We want to help every car in the future to be as autonomous as possible so that it can be as safe as possible. But we don't want to be a car company. We would like AI to advance as safely and as quickly as possible, as capable as possible. But we really don't want to host and provide a large language model service. And so notice in many domains and many industries, as you called it, we don't want to be the industry leader. We want to enable the industry to have leaders, and so that we could focus on our unique contribution. And so I think that we could play a really great role at that intersection in the three pillars that we mentioned. And you have such deep domain expertise. You have such passion for the methodology, and you have a pioneering spirit. You know, you want to go make this happen. And so, you know, I love people like that. I love endeavors like that, and I love you guys because of it. I think it's great.
Jensen, speaking of partnership, we built together BioHive 2. Our team was sleeping on the floor of the data center to get this thing stood up in 3 weeks, and your team was too. So kudos.
Where I stopped listening was, I was going, huh? Our teams were sleeping.
They were working hard.
My internal knee-jerk reaction went, huh?
No sleep. We stood that thing up in 3 weeks. And it is now the fastest supercomputer in biopharma. Is it surprising to you that the fastest supercomputer in biopharma is being built and operated by little Recursion and not one of the massive biopharmaceutical companies?
Is it actually in here? Is it Dick's?
No. The supercomputer, we thought about hanging it from the ceiling, but ultimately we had to move it to a data center down south, a few miles away.
When I walked in, I was thinking, now it makes sense. This is where our supercomputer is. NVIDIA was the first chip company to build supercomputers for our own use. Turned out to have been a good decision. Tesla is the first car company to build supercomputers. Not a bad call. Lots of other examples. The simple idea is whether we believe, you know, whether a company believes that in order to discover knowledge out of something where principled simulations will simply not get us there. You know, remember, there was a time when we thought molecular dynamics, with enough supercomputers, we're going to simulate the human body. We kind of gave up on that idea. We have a better idea. Isn't that right? And so there was a time where we thought, surely there was a time when I thought, that we're going to have supercomputers large enough to be able to do weather simulation to the minute to every region in the world within, you know, kilometer or a few hundred feet. It's now very clear that that's not going to happen. That even with NVIDIA's accelerated computing systems, it'll still require something 1 billion times larger, and 1 billion times larger is still a few decades away. And I'd like to see it in our lifetime. And so the way we solve that problem, and just to give you an example, in our world of computer graphics, we do this thing called ray tracing. And we thought ray tracing was still going to be 3 decades away. 3 decades away. Today we path trace, which is more advanced than ray tracing, everything. And the way we solved that problem, and the performance is incredibly good, and the way we solved that problem was we simulate 1 pixel, and we use AI to guess the other 64. Okay, and we're doing the same thing with climate. You know, we understand the physics. You don't have to go brute force simulate the physics for every single protein and every single cell. And so it's very clear we don't have to simulate the physics. We understand the physics of weather. We don't have to go simulate the physics for every square kilometer. And so we could teach an AI how to predict physics and let the AI go and tell us what's going to happen. Not because we want to understand the causality of weather. We understand causality of weather. We just want to know what the weather is going to be like this afternoon. That's all. Do you understand what I'm saying? That's a fundamental difference. That's a giant mental leap. That's we understand the causality of things. We want to understand how it's going to impact a person or some disease or whatever. How do we cure certain disease? We probably understand the basic biology behind it. Of course, the complexity of multi-omics makes it hard. We understand biology. It's just that in the larger scale of things, it is so hard to understand its interrelationships. We now need to use some other type of algorithm to go understand it, which is AI. And so I think that if you believe in these things, these principles, then you've got to, and then the second thing is if you believe that your company is fundamentally about intelligence, that your company isn't producing drugs, you're producing intelligence that leads to the production of drugs. And if this is how intelligence will be amplified, intelligence will be discovered, intelligence will be amplified and put to use, then how is it possible that you don't have the instrument of intelligence? And so when you think through it through first principles, the people who are able to think like that are able to make that next leap. If this is to be true, even though nobody's ever done it before, if this is to be true, and you fundamentally believe in something, and even though nobody's ever done it before, it doesn't mean you shouldn't do it. This is exactly the reason why you should do it. And so that we reason about things, in this way, and I think I know that most pioneers and leaders, innovators think in this way, and you guys think in this way. I think it's really terrific.
When you think back to how you thought that way 30 years ago, with the founding of NVIDIA, we've got analysts, investors, patient advocates, and also many founders, and I would say probably among our employees here, some future founders as well. You faced a lot of skepticism, and yet the first principles told you the direction to go. How do you, as a founder, as an early employee, as a visionary and a pioneer, how do you have that faith to keep building across multiple decades in the face of all of that skepticism?
Well, first of all, we reason about things from first principles the best we can. And sometimes the first principles don't exist because we either don't understand it completely, and maybe there's a bit of a leap of faith. You just believe it. It must be so. And all of that belief or that reasoning came from somewhere, some external information or you're informed about it somehow. If the facts don't change, why change your beliefs? If the facts don't change, we're a universal function approximator. You and I were both this giant deep learning model, and if the input doesn't change, why should the output? And so I'm kind of wired that way. I challenge my reasoning constantly. In fact, almost literally every day. And I gut check on how we arrive at a lot of the assumptions that we arrive at, because of a lot of people are at stake, and a lot of the work that our employees do are at stake, and so you have to reason about these things, but you have to double check your assumptions all the time. If things don't change, why do you stop believing in what you believe in? So you got to believe in something. And so you want to be as informed as you can. You want to be as reasoned as you can. You want to go back to first principles wherever you can. And then lastly, there's always that leap of faith that is necessary for all great things to happen. If it was obvious to everybody, then it would have already been done.
Somebody's got to go do it.
Somebody's got to go do it. And so So we believed in the first principles that general purpose computing, this universally general tool, can't possibly be the right instrument for every type of computing. And if you look at a CPU, for example, the ALU is doing the work. And that ALU is only a few percent. It's like a company where the engineer is doing the work only is like 3%. The other 97%, management overhead. Now, that's what general purpose computing is, and our first principles doesn't make any sense. And so we weren't trying to replace general purpose computing. We were thinking, there's a reason why CPUs exist, and we designed plenty of them. And it's good for whatever it's good at, but why don't we just augment it with something else? We call it accelerated computing. That's why we don't call it parallel computing. Parallel computing is kind of like, as opposed to sequential. The two of them kind of, you have code that's single-threaded, you have code that could be multi-threaded, And so we reasoned from first principles that if we were to figure out a way to get that to be the nature of computers, and every computer has those properties in it, and that there was a good programming model to deal with this kind of a complexity, then we can really change computing. And so we believed it from the very beginning. The question is, how do you find those applications? And we found one after another. The first one we found was computer graphics. That was a very sensible one. And then the next one was image processing, and then molecular dynamics, and particle physics, and fluids, and so on and so forth. And we just kind of kept finding our way there. And then one day we found deep learning. And so
it's incredible. Last question, because I know you have to go.
I don't have to go right now.
Okay. Then I've got 20 more questions.
Okay, good. Good. I'm happy to be here.
When we first met, 18 or 24 months ago, you told Ben and I something.
Chris is hosting all of you here. I need to fly back to host. I'm still working for a living too.
You told Ben and I that the natural state of every company was dying, or that every company was 30 days away from dying.
Yeah.
And that resonates deeply with probably every founder of a startup.
Yeah.
But hearing it from you, I think, could be surprising to people, and probably is also because you feel that one of the reasons NVIDIA has been so successful. Where's that philosophy coming from, especially at the stage of company you are now, and what can all of us learn who are building earlier stage companies from that philosophy?
I think NVIDIA's a supremely confident company, and you have to be in order to do great things. But you can't be complacent. And so somewhere between confidence and insecurity, maybe that's what it is, is where we live. That we always believe that we could do amazing things, but if we don't keep doing amazing things, we don't get to do this anymore. And so when I wake up in the morning, and this morning is no different, all these thoughts of problems, that's the first thing that comes to my head is problems, and the problems that should have been solved last night that wasn't solved, and so and so I wake up to those, and so good morning, and the problems are challenging, and I consider them somehow, for some reason, I've always internalized them to be existential. That if you don't take care of these things, eventually you'll get bad, and everything will come to an end. I felt that way in 1993. I felt that way this morning. I don't know how to. I think I need therapy. That's what it is. No, I'm just kidding. I don't think it's. I think it's good to be able to be a properly functioning adult. Both being confident you can achieve something incredibly hard that nobody's ever done before, on the one hand, and on the other hand, really having a sense that you have to keep working at it, and never be satisfied, and don't be complacent, and all of those feelings I don't think you want settling in, and so a long time ago, I used to share our financials. Well, I still share our financials with all of our employees. They're just public now, but back in the old days, I would share our financials with our employees every Friday. and I had a laptop, and I just, put it up, and we're looking at a spreadsheet, and somebody said, so Jensen, that our cash runs red in June. I think it was June. And I said, yeah, if we don't make money before then, we'll be out of business. The second question was, what does that mean? The engineer asked, so what does that mean? I said, well, it just means that if we don't, make more money in 30 days, we'll be out of business. And then I literally just said what that means. I mean, that's what it means. I wasn't trying to judge or anything. I just said, hey, what does that mean? I said, in 30 days, we'll run out of money. And then somebody else said, so what are we going to do? I said, well, don't run out of money. And so we have one of two choices. We can either make some, or we can go raise some. And I'm not sure we're good at either one of them at the time. And so I think one of the genuine possibilities is our company could go out of business. And we didn't. We always, somehow there's always a diving catch.
Spoiler alert.
Yeah, we didn't. And over time, I just want to remind people that if we don't do our jobs, we'll be 30 days from going out of business. And then all the employees that were there at the time told all the other employees that we have to really stay focused, because if we don't, we could really go out of business in 30 days. And then one day we had gone public, and we're having a company meeting, and somebody asked, Jensen, we heard rumors that we could be out of business in 30 days. And I said, hey, listen, if we don't do our jobs in the next 30 days, we could go out of business someday. So yes, we could be out of business in 30 days. And so all of the new employees just panicked. But anyways, I think your psychology should be that as a startup, every day matters. And I can tell you with great confidence, as a startup company, every day matters. And so the next 30 days, the decisions you make, the things that you do, the people you hire, the accomplishments, the culture you create, all of those things, all of those, they could lead to either you being vibrant, succeed for a little longer, or go out of business in 30 days. I completely believe that, and I believe it for me. And so.
Thank you. And the corollary, I think, to that in our industry is that we get to know some of the patients who we hope to one day be able to treat. And those patients are waiting, and so every day matters.
Every day matters, yeah. Every day matters.
That mission.
Yeah, every day matters. And look, we're volunteers. All of you are volunteers, 100%. 100% of you are volunteers. You don't have to work here. You don't have to work, for many people, you don't have to work at all. And so you surely don't have to work here, so you're volunteering to work here.
But please keep working.
No, yeah. You're volunteer to work here. You're volunteer to work. Nothing is stronger than the heart of a volunteer. You're volunteer to work here. And the reason why you volunteer to work here is because you believe in the mission, you like the people, you enjoy the people, you believe you're doing something important, and so you get to do it every day. If suppose everything I said was true, then you get to do it every day. Why wouldn't you want to do it every day? So do it every day. That's the way to think about it. So instead of, it's not because we're obligated to work every day, or gosh, we got to do our best work today. We get to do our best work every day. And so I think you guys are, you're during just an extraordinary time in your industry.
Yeah.
And you're also going through an extraordinary time in industry. You have to realize that the instrument of your work has not been reinvented, reimagined in 60 years. The year after my birth, IBM announced the System 360, the general purpose computer architecture that's remained largely the same until now. And all of a sudden, we created this thing called accelerated computing, and on top of that, a new computing model called generative AI, and it's changing completely everything. how software is written, how software is processed, the type of software we can write, the type of problems we can solve. For the very first time, completely revolutionary. It's changing every single industry. And so for us to be, us as an NVIDIA, to be at the center of this industrial revolution, this computing revolution is extraordinary. But for you to be able to utilize this capability to revolutionize one of the most important industries of all, humanity. The timing is incredible. This is a once-in-a-lifetime opportunity for all of you. This is a once-in-a-lifetime company and a once-in-a-lifetime circumstance. And so if you know, boy, if you could slam 2 days into one, I do it. You know, so this is just such a fun time for you guys. I'm here to tell you I'm jealous. You know, this is really, really great. I'm very proud of you.
We have an opening if you're interested.
What, do you have an opening as a patient?
I hope not.
Okay.
I hope not. So thank you, Jensen.
Yeah.
This has been amazing. Before we go, at GTC, your conference, a member of our team was wearing something very special, which was this rainbow GPU necklace, which you signed, and we have literally encased.
Wow.
And you asked us there to make you one, so we did.
Oh,
this
wow.
This is your rainbow GPU necklace.
Okay, I'll wear it.
And you also asked us to make 2 for your kids. And so, big thanks to Jenna and the team.
My
who
gosh,
put these
thank
together.
you. Thank you so much. This is
it's an early architecture.
How many teraflops is this?
I don't know
How
if
many
Imran's here.
teraflops is
This is one of the earlier ones.
Okay, this is beautiful.
But big thanks. And would love, before you go, just to get a picture of you with
Yeah,
everybody
I would love
here.
it.
Is that
I would
okay?
love it. Thank you.
All right, thank you so much.
Thank you so much for coming.